Hello and welcome, Doffy, Bob Marley, Bill, and Cernu. Always a pleasure to have you pass by. Hope everyone's doing well, and also thank you so much for passing by. As usual, Corey Campbell, and well, I guess maybe just some updates like this week or this past week. Definitely a rough week. Schedule did not go according to plan, but that's okay. That's life. That I've learned a long time ago, that things don't go according to plan. Where, for whatever reason, my sleep definitely not holding up for reasons that I can't explain. Really trying to emphasize sleep hygiene days. So far, it's not paying off, but I'm hoping at some point it will pay off, because I can't really explain what's going on with my. I've cut out caffeine. Not that I was a huge caffeine drinker beforehand. I would usually just have some caffeine in the morning, then that was it before my workout. That is off the menu for me for now, trying to eliminate all variables to get my sleep sort of under control or whatever it is. Because it is just really bizarre and something I've never experienced before, or at least for this amount of time, which is super frustrating. That's okay. I will figure it out at some point or another. And a couple of other life things getting in the way. But all that to say, I'm back. Well, I'm here. I planned to stream yesterday, but obviously that didn't happen. And even though I didn't get the greatest sleep last night, got just enough to find the energy today. Because there's this paper that was sent to me by a fellow community member, Desperado Sensei, which I super appreciate. And shout out to them, where they wanted me to cover this paper, and they sent it about a week ago, and it coincides pretty much right in line with me getting my tablet, which I now have, and I think I have it set up. This is going to be the first stream where I try to incorporate it. Might be a massive fail. Who knows? We'll see. But I'm excited to give it a try. And the paper that was sent to me, I think it's a good trial run. How I can incorporate drawing figures and getting concepts across drawing figures, because it's a paper that's biochemistry / molecular biology. It's not super long and lengthy, and it's pretty much right sort of in my wheelhouse, or maybe wheelhouse adjacent, where it's mostly molecular focused. Or actually, I think it's actually a good mix of both molecular and biochemistry. I'm actually kind of excited to get right into it, because I think it's really cool. And I had to set aside some time to actually sort of think about, okay, how am I going to approach this? And I had to do some other readings, because this research, so even though it's right in my wheelhouse, I didn't understand everything upon first reading. Whereas other research that I had to dive into, basically this research group was building off of past research, and they pretty much have published a series of papers kind of leading up to this one, where past research set up the groundwork or the foundations, and then this is just them continuing on in that line of research. And when I first read the paper, it's okay, I more or less overall understand the concepts, and I know the overall main messages, all that stuff. But then I had to ask myself, okay, how do I actually explain everything that's going on here? And at that time, after the first reading, I definitely did not understand everything that they were doing. I had to go back to the previous publications and see what they were doing there. And now I think I have a much better grasp of more or less exactly what they're doing. And what they're doing is actually pretty cool. As alluded to in my stream title, they're studying evolution, but they're studying evolution in vitro. And the experiment or the overall system that they've designed to do that is actually pretty cool and elegant. And hopefully I can get that across once I start drawing things out and explaining it, because you have to know, you don't have to, but helpful to know about RNA, it's helpful to know about DNA, some of the fundamentals about DNA and RNA and how those are replicated, etc., etc. So I want to make sure that I set enough time aside to do that. I'm still sort of uncertain. Do I dive right into the paper, or do I go into the past research first and then the paper? I think I'm probably going to go into the past research to set the foundation, and then dive into the research to like pretty much dissect that paper, which is not super long. It's like 3 or 4 pages, and it's I would say really elegant and really cool. But that might just be my opinion. But getting right into it. But before that, just covering this one thing, because I want to make sure I actually have enough time to cover what I want to cover about that paper. I'm going to cover this and get into the research I'm going to cover. Because I think this is important. Where this was published a few days ago, where NASA announces summer 2023 hottest on record. So, summer of 2023 was Earth's hottest since global records began in 1880, according to scientists at NASA's Goddard Institute of Space Studies in New York. The months of June, July, and August combined were 0.41 degrees Fahrenheit, or 0.223 degrees Celsius warmer than any other summer in NASA's record, 0.1 degrees warmer than the average summer between 1951 and 1980. August alone was 2.2 Fahrenheit, or 1.2 degrees Celsius warmer than the average. June through August is considered meteorological summer in the Northern Hemisphere. This new record comes as exceptional heat swept across much of the world, exacerbating deadly wildfires in Canada and Hawaii, and searing heat waves in South America, Japan, Europe, and the US, while likely contributing to severe rainfall in Italy, Greece, and Central Europe. And here in my area, Quebec, I would say this, at least from my perspective, I didn't find the summer, like, we definitely had heat waves and brief heat waves, but they're not super extreme. And it seems like I'm not going by any sort of data, this is just like my subjective experience, but the summer seemed shorter, but like pretty much it was like short, intense moments of heat, and then like pretty significant cooling, as in like I would say this summer's overall, from my subjective experience, unseasonably colder than usual. But then, as an example, like literally a week ago, or a little over a week ago, it was like 40 degrees Celsius here for like an entire week, which is like unheard of, at least in my experience in September. But then like this week, it's pretty much, we're officially in fall. It's like, as of right now, it's like, 15 Celsius. Like we pretty much had like an almost like full, 20-25 degrees Celsius swing in the past week, where, yeah, it was really strange going from like 40 degrees Celsius and like now, like almost overnight, I just like don't need my air conditioner. Oh, and Afflin Pogform, welcome. Thank you so much for passing by. Great to see you. So it's a great question. And also, thank you for posting all the great research that you posted in the Discord. That was great. Definitely a lot of interesting research. But as mentioned, certainly some of it outside of my area of expertise, but totally looks super cool. But 40 degrees Celsius, I believe, is like 110 Fahrenheit, maybe 100. I could be wrong. No, maybe like 105 Celsius to, yeah, 104, 105. And I assume through like satellite data, but I don't actually know how they're recording it. But summer 2023's record-setting temperatures aren't just a set of numbers, they result in dire real-world consequences. From sweltering temperatures in Arizona and across the country to wildfires across Canada and extreme flooding in Europe and Asia, extreme weather is threatening lives and livelihoods around the world, said NASA Administrator Bill Nelson. The impacts of climate change are a threat to our planet and future generations, threats that NASA and the Biden-Harris administration are tackling head-on. NASA assembles this temperature record, known as GIS temp, from surface air temperature data acquired by tens of thousands of meteorologists. So this is, so they're getting into how they're collecting it. Not satellite, it's stations across the world. meteorological stations, as well as sea surface temperature data from ship and buoy-based instruments. This raw data is analyzed using methods that account for the varied spacing of temperature stations around the globe and for urban heating effects that could skew the calculations. The analysis calculates temperature anomalies rather than absolute temperature. A temperature anomaly shows how far the temperature has departed from the 1951 to 1980 base average. Exceptionally high sea surface temperatures, fueled in part by the return of El Nino, were largely responsible for the summer's record warmth, said Josh Willis, climate scientist and oceanographer at NASA's Jet Propulsion Laboratory in Southern California. El Nino is a natural climate phenomenon characterized by warmer than normal sea surface temperatures and higher sea levels in the central and eastern tropical Pacific Ocean. The record-setting summer of 2023 continues a long-term trend of warming. Scientific observations and analyses made over decades by NASA, National Oceanic and Atmospheric Administration, and other international institutions have shown this warming has been driven primarily by human-caused greenhouse gas emissions. At the same time, natural El Nino events in the Pacific pump extra warmth into the global atmosphere and often correlate with the warmest years on record. With the background warming and marine heat waves that have been creeping up on us for decades, this El Nino shot us over the hump for setting all kinds of records, Willis said. The heat waves that we experience now are longer, they're hotter, and they're more punishing. The atmosphere can also hold more water now, and when it's hot and humid, it's even harder for the human body to regulate its temperature. How come the US uses the I would say, arguably the US is way behind every other developed country when it comes to how they measure temperature. Fahrenheit, I don't know what that system is. Willis and other scientists expect to see the biggest impacts of El Nino in February, March, and April 2024. El Nino is associated with the weakening of easterly trade winds and the movement of warm water from the western Pacific toward the western coast of the Americas. The phenomenon can have widespread effects, often bringing cooler, wetter conditions to the US Southwest and drought to countries in the Western Pacific, such as Indonesia and Australia. Unfortunately, climate change is happening. Things that we said would come to pass are coming to pass, said Gavin Schmidt, climate scientist and director of GISS, and it will get worse if we continue to emit carbon dioxide and other greenhouse gases into our atmosphere. NASA's full temperature dataset and the complete methodology used for the temperature calculation and uncertainties are available online, which is great that they're sharing their dataset, although I'm definitely not in any position to analyze it. And the GISS is a NASA laboratory managed by the Earth Sciences Division. Okay. So yeah, it's not... So I was totally wrong on my speculation. Not satellite data. Basically, I actually probably should have known, because I literally have a story about stations across the world. But like, yeah, it seems like pretty much meteorological stations and, buoys and things in the ocean. Basically, I guess. How do they call them? Yeah, buoy-based instruments. And also, I guess, actual ships in the ocean. So they are actually sampling actual locations on the Earth. Temperature at those locations. Makes sense. It feels like summer was warmer everywhere except where I'm at. Well, I think that's one of the whole issues with climate change. I guess maybe one of the reasons why we moved away from global warming, because it's not exclusively, universal warming at all times. And true, not yet, at least. But yeah, like I said, here this summer, there were definitely moments, like stretches of weeks, where it was like super unseasonably hot, but then there'd be weeks or stretches of weeks where it was just like unseasonably cold and like lots of rain. And like I said, a good example is that is just like, in the past like 2 weeks, went from like ridiculously hot, like just like, literally. I mean, I canceled pretty much all streams last week because of the heat, and like I didn't want to imagine streaming in the absence of my air conditioner. You'd just be watching me like sweat. And that did not seem appealing at all. And now, like, yeah, this week, I literally just last night uninstalled my air conditioner. I saw like the projected forecast, 15 Celsius, or it's not supposed to get super hot anymore. like pretty much overnight, just going from straight like summer to fall, like a hard transition. I don't remember that occurring in the past, but I could be misremembering. I'm not going by any hard data, my subjective experience. Probably gonna cover this later if there's time. One second. I want to dive into actual research. Okay. So yeah, this paper, published 2009. I actually did not check if there's follow-up research. I actually should probably do that. But yeah, it was sent to me by a community member, Desperado Sensei. Niche partitioning in the coevolution of two distinct RNA enzymes. And they wanted me to cover it, which I was more than happy, because once I started reading it, it's like, oh, like this actually super interesting. And also, like, again, pretty much right up my alley in terms of like biochemistry, molecular biology. It's out of the departments of chemistry and molecular biology, and the Skaggs Institute for Chemical Biology, the Scripps Research Institute. So, a pretty, generally speaking, world-renowned research institute. And yeah, it's like, 4 pages, maybe 3 pages, excluding the methodology, materials and methods. And, obviously some of it, some space is taken up by figures. So I think it's a good starting point of basically testing out how I can approach, drawing figures and getting concepts across, drawing stuff out, if I can actually do that, now that I have the capability to do that. But before getting into this, so yeah, once, as mentioned, I started reading it, I overall understood it, and I overall understood, like, the methodology, what they were doing. But to really understand, like, all the nitty-gritty, I had to go into the past research, because in, like, the second paragraph here, they describe something, and they describe something specific, but what they're doing. And I totally could not follow it. Which, that might be a me issue, but, I know this from other researchers where totally it happens where researchers read research that's in their field, or, looks like in their field, and, there's still challenges and struggles in understanding things. Because it's just the nature of the game. And it's a testament to how difficult it is to read research. And that, to read research, like, even though I'm comfortable with, I think, a pretty wide range of research, there are challenges that I encounter where, yeah, something can be, like, right in my field, and I'll have, as exemplified here, difficulties understanding it. So I have to go back into the past research that they have done. So it's by 2 authors, or, like, grad student and, main author. And they have past research here. So that was 2009. This is 2007. And pretty much it seems to be like their specialty. And they have this other past research, also including this same person, Harold F. Joyce. That seems to be like the main player. They are probably the one that's, the head of the research. And yeah, I had to pretty much, like, go back and, like, look at these figures. So, like, this figure that I'm hoping to dissect and explain what's going on here, and actually use this to get the concepts across. This was left out from this research, which, not necessarily a problem, because they describe it in words. I think they're probably banking on the fact that, if the experts in this field, they are likely following this research, and so they're already familiar with this. And they use this figure multiple times, even in this paper here. This is in 1997, so it goes back over 20 years. And it's just an updated version of this schematic. And, it looks intimidating, and, like, at first glance, I could not understand this, or at least when I saw this, it wasn't like, oh, I know what's going on here. Like, I really had to, take my time and really, try to make sure that I know exactly what's going on here. And even now, I can't say with 100% certainty that I know exactly what's going on here, but I'm highly confident because this is just pretty standard molecular biology. You must have heard about that recent kerfuffle with a climate change paper. I believe I know what you're referring to. Yes. I do cover retractions. I do cover what I think is poor quality research, or, research that has been deemed poor quality by other experts in the field. I'm definitely not the arbiter of, what is good or bad or poor quality or good quality research, unless it's like right in my. I've used AI to so not yet, and actually that didn't even cross my mind, to be perfectly honest. I should maybe test it out. I'd be kind of skeptical, but that's just my bias of being a skeptic of AI and its ability to, like, is AI gonna give me like the tools to understand this figure? Maybe it will. Like, I'm just totally thinking out loud. I don't actually know. But I suspect it won't. And that's really, I think, one of the barriers here. But I should, if you want to send me some plugins or so as someone who I trust, if you want to give recommendations of AI, by all means, send them to me. I'll check them out. And totally molecular bio is hard to read. For oh, Chris Whoppers, welcome. Great to see you. So I'll have to plead ignorance on Casimir cavities. But now I'm curious. Not sure I've heard. Looks like oh, okay. This is like physics, or I guess quantum mechanics to be more specific. Which, yeah, that is definitely as cool as I think quantum mechanics and physics and all that stuff is. Totally, the most I can talk about is I know van der Waals, but that's mostly like a chemistry thing and, like, molecular forces that I can talk about. But overall, quantum mechanics. So yeah, all that to say, I do not understand supercavitation very well. I'm not even sure I knew what supercavitation was. He brought it to my attention. Yeah, I have no doubt that it's interesting. Like, everything that I read, assuming I understand anything that I read about quantum mechanics and quantum physics, sounds very cool and super interesting, and stuff that I want to, like, dive deeper into. It's just that, well, like, yeah, since it's not my area of expertise, it's a larger barrier to entry. GPR has a data analysis thing now where you can upload an Excel sheet and it will do whatever graphs. Oh, yeah, that doesn't surprise me. So, actually, the funny maybe, I don't know if it's whether it's funny or not, but yeah, the funny or interesting thing is like the hard data, like they the numbers that they're going to report. I understand the numbers. Like, I'm going to try to explain what KCAT and KM are in the context of, like, enzymes. So, like, enzyme kinetics and, catalysis. That's pretty much, as biochemistry as you can get, arguably. And so, like, and these are, like, concepts that I teach or have taught to students. And so, like, I know exactly in terms of, like, the data that they generate, I can interpret that. But it's more like, how are they generating this data? Like, what is the actual experiment that they're carrying out? And the biggest burden is this figure here. Understand how are they? So what they're doing is they're setting up evolution in a test tube, so to speak, or as in the stream title, in vitro evolution. So we should probably just, like, start, and I should hopefully try to explain as we go. Once we reach a roadblock of what I think should be expanded upon. But basically, as they're going to highlight, it's evolution is a thing, whether you want to believe it or not. That is the foundation of all of biology, and explains all of the diversity of life. And we have pretty good understandings, or very thorough understandings of evolution. But do we understand absolutely everything about evolution, right down to the molecular level, or like the deep molecular level? No, there's still sort of maybe not unanswered questions, but I would probably more frame it as details to add in of like, we know what's going on here, but we don't know the exact how of what's going on. And one might ask, well, why study evolution in vitro? Well, one of the advantages is that, or maybe not, I shouldn't start there, but one of the burdens of studying evolution is like evolution takes place on, long timescales. And evolution, occurs generation after generation. As in, what matters and what generates genetic diversity is, from one generation to the next. And so, let's say, for example, if we want to study, human evolution, okay, well, first, we can't go back in time, not yet at least. And two, even if we wanted to start now studying human evolution, well, then we need to wait generations. Okay, and that takes, decades, centuries. So the question then becomes is, can you study evolution or, get a better understanding of evolution if you can somehow design a system that will pretty much generate or create lots of generations in a short amount of time? As in, to use the human example, can you generate multiple generations of humans in, less than a year? Okay. And that is pretty much why there are attempts to study evolution in vitro. I guess a good example is E. coli. As in, E. coli is a model organism for a variety of reasons. Simple bacteria, ubiquitous, largely harmless, pretty much majority harmless. And one of the advantages of E. coli is they have a short generation time. As in, from one generation to the next, it's like 20 minutes, 30 minutes. Yeah, they divide every 20 minutes. I use E. coli, I study E. coli, and this is for a variety of reasons. One of the reasons why E. coli is a model organism, because they just duplicate so fast. And so, E. coli has been used to study evolution, because, like, yeah, as opposed to waiting for 3 generations of humans, and that might take, 50, 75 years or longer, to generate 3 generations of E. coli takes an hour, hour and a half, right? So you can way more quickly study genetic transmission and, evolution on sort of an accelerated time scale, so to speak. Now, there are all these obvious drawbacks in that, you're using E. coli and, you're limited there, and there might be some obvious challenges in how it extrapolates to, humans or, other organisms. But that's always, the trade-off in science. There's never going to be that perfect system, at least not yet. Do I understand bioengineering yeast to create any compound, basically selecting specific enzymes? Absolutely. That is so. You're essentially talking about synthetic biology, and actually, at my institution, like there's a company, sort of a biotech startup, that is essentially trying to use yeast to generate cannabis compounds. Basically, it doesn't get any more simple than that. Of like, instead of growing cannabis or all using plants or getting cannabis products from plants, you can just potentially, well, that is what this company is trying to do. Of like, you can genetically engineer yeast to produce all of the cannabis compounds and just purify them there from like a bioreactor. I feel like the data on our appendix efficiency over generations is a good set for evolution applications. Also mushrooms. Yeah, that's true. I don't know. I imagine mushrooms probably have a longer generation time than I'm pretty sure they do actually. Yeah, like it's hard to compete with 20 minutes, but here it's even less than 20 minutes. Octarine Bio does that only. Yeah, there's a bunch of startups. I know of this one company because it's like pretty much affiliated with my academic institution. But like, yeah, there are a bunch of companies that are trying to get into the space of like, understandably, as hopefully cannabis gets more and more legalized across the world, there's going to be a greater demand for cannabis products. Not only just whatever cannabis plant and whatever flower, but CBD and all the other cannabis related compounds. And so it's not efficient, or it's not that efficient to grow a cannabis plant. And we're not interested really in like the entire plant, right? We're only interested in like very specific compounds in the plant. And so yeah, like there are multiple research efforts, if not like probably thousands across the world, basically genetically engineering yeast or other organisms to generate a whole host of compounds from, as you mentioned, Chris Whopper's psychedelics, to all of the wide variety of cannabis compounds that are, cannabis plant. And yeah, you're absolutely right. Mushrooms are always doing weird stuff. So they're going to set the stage, and we're going to read this paragraph, and then it's probably going to require more explaining. And this is where I'm going to have to like probably, or almost certainly draw things out. What are they describing here? Because even when I read it, I was like, I don't know what's going on here. So one of the aims of laboratory evolution is to model biological evolution experimentally with precise control of the key variables relevant to processes of selection, amplification, and mutation. Several laboratory evolution systems have been developed in which populations of bacteria, viruses, or macromolecules are challenged to adapt to various selection constraints, such as depletion of a critical nutrient or alteration of the environment. The use of catalytic RNA in such evolution experiments is of special interest because of the relevance of RNA to the origins of life and the simplicity of representing both genotype and phenotype within the same molecule. So, as they highlight, many systems have been explored or used to understand evolution, or to tease out more details in regards to evolution and how it occurs at every level, from the organism to the molecular level. And they refer to these other systems, bacteria, viruses, macromolecules. And yes, we can potentially use things like mice and other animals to study evolution. But as I already mentioned, there's drawbacks in terms of generation time, and also there's challenges when it comes to how much control do you have over the environment, or like if you want to control every single variable, or almost every single variable, then pretty much like the in vitro way, or like bacteria, viruses, and using that, you have way more control of all the variables and the environment at a molecular level. And again, that's just another trade-off. Yeah, you can use animals, but you have less control. But animals more relevant to humans. But then here, going in sort of like the complete opposite direction, it's a trade-off of like, well, we're looking at pretty much a molecular system, but we have like precise control of virtually all the variables. And so we can do like fine tweaking to see exactly how evolution is playing out, or how natural selection is playing out. That is actually like what they do. They almost, arguably, if not maybe in reality, like they're watching natural selection occur in real time, at least based off of how they describe it. Of like, oh, in this test tube, we're seeing natural selection like play out as one species goes extinct. That is actually what they'll describe, and that's by design through their system. And their system is, once I understand it, hopefully I understand it. I'm confident that I do. It's actually like quite elegant and like quite cool. And they describe their system here. But let me just catch up with chat in a moment. MIT found that 20 millisecond pulse rates of ultrasound between 25 megahertz and 100 megahertz completely ruptured the COVID-19 spikes in a fraction of a second, rendering it acellular debris to metabolize naturally. Yeah, well, so that's actually a known thing. I mean, that's just sounds like sonication. I could be totally wrong. But sonication is like a known thing to break, cellular membrane. So wouldn't necessarily surprise me that, like, yeah, you can pretty much use sonication to, with the right frequency, as you sort of highlight, you can destroy a protein, even the spike protein. But yeah, sonication is totally a common thing. There are many ways to destroy, or to use the technical term, to lyse a cell, but that just means fancy terminology for rupture the cell and release all the cellular contents. So, what is their system? So that is a valid question. I would say genetic drift, not really all that applicable in this research. But it's totally like a valid question. But that's maybe a little bit beyond the scope of what they're interested in. A totally good question. So one especially powerful system, although it's funny, they're kind of patting themselves on the back because like this is a system that they designed. Like they're referring to like this system that I'm going to describe, but it's designed like by them. So they're pretty much like saying our especially powerful system. For the evolution of catalytic nucleic acids, involves the continuous in vitro evolution of RNA enzymes with RNA ligase activity. And just bear in mind, I'm going to expand more of what exactly is. Also to touch upon this RNA aspect. So as they end here, the use of catalytic RNA in such evolution experiments is of special interest because of the relevance of RNA to the origins of life and the simplicity of representing both the genotype and phenotype within the same molecule. So they are using and highlighting RNA specifically for a variety of reasons that they touch upon. One of them being that, I haven't looked into, the most recent hypothesis of the origins of life, but I know, whatever, one of the current ones or contemporary ones is, RNA as the first replicating molecule, as in the origins of life may be RNA based, and RNA is a nucleic acid, and so I think most people are familiar with the genotype and phenotype, but basically genotype is just, your genetic information, and your phenotype is, you can essentially think of it as, how you look sort of like visually, or pretty much like the behavior that stems from the genotype, as in, like, I guess to use the human example, skin color is the phenotype, but that's rooted in genetic information. But so there's a little bit of a sort of a disconnect in terms of like going from genotype to phenotype of like, yeah, the gene has to be expressed and then, a variety of things have to happen to get the phenotype that is my skin color. As opposed to in RNA, as they highlight, you have genotype and phenotype within the same molecule. As in, as you are changing the RNA, you are not only changing, the nucleic acid sequence, the genotype, but you're also changing the phenotype. As in, it's pretty much like one and the same, as opposed to not the case for potentially other organisms. Or like genotype is not always linked to phenotype in an organism. Where, yeah, using just basic humans as an example, you have your genetic information, your genotype. That gene has to be transcribed into RNA, and then that RNA has to be translated into protein, and then the protein goes and does its thing, depending on what protein it is. And so there's multiple steps in that process, as opposed to RNA, that is not occurring. It's just like the RNA is there, it's composed of RNA, and then pretty much the behavior of the RNA is tied to its genotype. They are directly linked. And so, as they say, that is one of the advantages of like pretty much using this system, where pretty much genotype and phenotype are inherently linked in an RNA world. So, in this system, RNA enzymes are challenged to ligate an oligonucleotide substrate containing the sequence of an RNA polymerase promoter element to their 5' ends. A primer complementary to the 3' end of the enzyme is extended by reverse transcriptase to generate complementary DNA, or cDNA. The cDNAs derived from reacted RNAs are then transcribed by RNA polymerase to generate many progeny RNA enzymes. Through repeated rounds of these steps, all carried out within a common reaction mixture, the population expands exponentially. Growth can be continued indefinitely by performing a serial transfer procedure, whereby each transfer provides a fresh supply of reaction materials and the opportunity to modify the reaction environment. So, when I first read this, it was like I sort of understand what's going on here, but I pretty much don't understand what's going on here. And so they have a reference here, 5. And so I had to go back to this reference because I hoped that they would expand on it more, or I can get a better idea. What they're doing is they are essentially describing a system that I'm going to dive deeper into. The only chemistry I like is aldehyde oxidation, basically forming new nitric acid adducts with aldehyde additions. This definitely brings you back to sort of organic chemistry days. Biology is very cool because it goes into interesting that does it go into? I mean, I guess physics is everything, right? That is sort of, I guess, the ultimate discipline. Once you zoom in close enough. In the physics community, they debate that DNA is a scalar, whatever that means, perhaps some sort of data transmission, as in scalar versus like vector. And yeah, it is. So that is pretty much one of the primary advantages of this, is that it's a fast reaction. So, what is their system? Or as they describe, what is their especially powerful system that they described for evolution? So, here is where I'm going to have to, if I can get this running. Whoopsies, sorry. If this works. It does. Okay, yeah. So I have this schematic. Good. All right, there we go. Okay. I should probably make it a little bit bigger. That. I can move. Okay. So this is their evolution in a test tube. And it's a cycle. It's very, once I understood it, assuming I understand it, which I'm confident that I do. Oopsies. Because this is just like strict molecular biology, or it's pretty much as molecular biology / biochemistry as you can get. This is catalytic RNA. So what's important to know is like this is a mixture of both DNA and RNA. As in the black lines is RNA, and the sort of white box is DNA. I want to make sure I have that from this page. One or... yeah, it's this one. Let's see. Yeah, RNA and DNA are depicted as solid lines and open lines, respectively. The open lines is the DNA, and the RNA is basically the solid black. So, going back. So pretty much the only DNA is like this here. And well, pretty much that is what they mean by the basically the not solid black line. Pretty much everything else is RNA. So you can pretty much think of it as most of the stuff is RNA, and you have little things or stretches of DNA. As in this right here is DNA, and they're using it. And they're using it for specific purposes. So what their system is like this molecule here, that is RNA, okay? And it's a catalytic RNA, or it's an RNA ligase. As in, all it does is it takes two RNA molecules and joins them together. Do I understand? Oh, DC Drunk, welcome. Thank you. So, I don't know about that, but I appreciate the sentiment. My family is all biochemists, and they talk extensively about telomere failure, but I thought it was chloroplast degradation. Well, maybe in plants, unless you mean, well, so chloroplast, sort of analogous, I guess, to mitochondria, humans. So, well, I guess I could never say that I understand aging. Arguably, no one fully understands aging, but I follow some of the aging research. And I know sort of some of the research about, telomeres, and sort of the multiple hypotheses put forth in regards to aging, but I'm definitely, like, not an expert. And from what I understand, but my understanding is limited, there's a little bit of debate, or, we're still trying to actually, like, tease out what exactly aging is. But I know that this telomere gets a lot of traction. But I don't, as far as I know, not, like, fully accepted of, like, this aging only boils down to telomere. I think it's pretty clear, and I think most experts agree that there is more to aging than just telomeres. But I know that there are many research groups across the world, like, studying, and I follow the research. Well, actually, maybe as a preview of future content that I want to cover, is this longevity factor. One of, I guess, the key things that we know with a high degree of certainty about aging is cognitive decline. There's lots of efforts put forth to how to reduce or hopefully eliminate cognitive decline as we age. This research is right up the alley that I plan to cover at some point. Well, I guess when it comes to, living organisms, yeah, I was gonna say, like, rocks degrade, right? As in, like, water erosion, etc., etc. So, that's arguably the aging of rocks. But yeah, so to describe their system, or to expand on their system, so this is an RNA ligase, and it just takes this right here. That is an RNA. So this is a combination of, like, RNA and DNA. And what an RNA ligase in this case, and that's like this thing here, that is an RNA. It just joins, two RNA molecules together. So that is what it does. And that's what they're depicting here in the first step, as in pretty much to go from, well, I guess pretty much like this arrow here. All it's doing is this is attaching this right here. That's all. And you can see, well, hopefully you can see, it's like right here. That is the RNA part. So it's just, you can think of it as stitching it together. And so it's just stitching two RNA molecules together. And you have this stretch of DNA hanging off. Okay? And that's actually important, because they use this DNA down the line. So once you have this new molecule right here, okay, it's pretty much the same as the original one, but you have this extra stretch here with, this small part of RNA, and then this small part, or this slightly larger part of DNA. So then what they do is they add in another piece of DNA, and this is a designed piece of DNA. Okay? And it's designed to bind to a certain stretch on this larger molecule here. And they do that for a reason, because they basically want to get it double-stranded. So, you have RNA and DNA. One of the primary differences between RNA and DNA typically is that DNA is double-stranded. Virtually everyone knows about the double-stranded, double-stranded helix of DNA, versus RNA is single. So what they do is this small piece of DNA sticks to, and it's designed to, stick, or I guess the important part is hybridize. That's what these little lines here represent. They represent sort of like binding, and binding to a specific region. It's designed in a very specific way. It has a specific sequence where it will only bind this location on the RNA. And then once that is there, okay, it's designed that way because this, okay, is a docking site, basically. It's a docking site for this other thing here called reverse transcriptase. And what's special about reverse transcriptase is it can make DNA from RNA. So generally speaking, if you talk about the flow of DNA to protein, this is where I can sort of like draw things out, or at least come up with some sort of like schematic. In, let's say, a human organism, you have your genetic code in the form of DNA, okay? That gets transcribed to RNA, and then that gets translated to, that's not how you spell protein. You pretty much go from DNA to RNA to protein. And as sort of highlighted by the arrows, it's pretty much, at least in humans, it's one direction. Go from DNA to RNA. That is called transcription. And then from RNA to protein, that is called translation. But there are some organisms in nature, because, nature does some cool and interesting things, where they have the ability to go in the opposite direction. Not from protein to RNA, since I know that's virtually impossible, not occur in nature yet. And even then, even through artificial means, virtually impossible. But there are some organisms go from RNA to DNA. And this is actually what the HIV virus does. And that's why it's called reverse transcriptase. If we go back to, the diagram, as I have it circled here, reverse transcriptase. It's called that because, as I mentioned, going from DNA to RNA, the process is called transcription. And so, obviously, to go in the opposite direction, they call it, no surprise, reverse transcription. And the enzyme in this case, they called it reverse transcriptase. So that's all that is occurring here. Is you want to eventually go back to DNA during this process, and then to go back from DNA to the original molecule here. Really, that's what it's depicted in a circle, so it's a cycle. And that is the ultimate goal, is to regenerate this original molecule, but with a mutation, okay? That is where the simulation of natural selection comes in, where pretty much all throughout this multi-step process, you can have mutations that occur, either naturally or kind of like engineered, without getting lost in the details of how do you engineer mutations, but there are ways to do that. And they essentially like do that at some point of like pretty much manipulating the conditions such that you introduce mutations. But at the end of the day, on a long enough timeline, mutations will happen at some point in this cycle. And so, yes, the ultimate goal is to regenerate this original molecule here, but through in enough repeated rounds, there will be some mutations introduced that will change it a little bit, and then through, tens of thousands, hundreds of thousands of generations, might change it, a lot. And so, yeah, once you have the reverse transcriptase, I should probably get rid of some of the things. Don't really need this anymore. So, yeah, this reverse transcriptase, even though I just erased it and can circle it again, we are going from RNA to a mixture of RNA and DNA, as you can hopefully see here in this molecule. Remember, the dark solid lines, that's RNA, and then the open box line, that's DNA. So here, you have both RNA and DNA in a single molecule. This is double-stranded. So pretty much what we want to get to is double-stranded DNA. And then this right here, this RNA polymerase, that's just, fancy terminology in molecular biology of like this will make RNA. But it will make RNA from double-stranded DNA. So what's important here is actually this part. So the rest that you can hopefully see is the solid black line. That is the RNA. But so under the RNA, okay, the other strand is DNA. So that's irrelevant at this point. What's most important? And Wicked Beta, welcome. Thanks so much for passing by. I appreciate, hopefully, the positive feedback. I am definitely covering some biology here. And so if you're a fan of biology, well, I think you'll fit right in. So yeah, well, the important part here is, like I said, the open box is DNA. So here you have, right here, double-stranded DNA. Okay, that is the key point that we want to get to, or that they wanted to get to. I'm not included in this. I didn't carry this research. And as you can see, you have these two words here. They're pretty much, it's shorthand for promoter. Okay, and this is just, molecular biology terms for, you can think of it as a docking site. Okay, it's a specific sequence. In this case, it's a specific double-stranded DNA sequence, okay? And it acts as a docking site, okay? It acts as a docking site for this specific enzyme here. I don't need to circle the whole thing. Actually, the RNA polymerase. So this RNA polymerase, it can actually recognize, okay, this docking site. That's what this promoter region is here. And then it can dock there and then make RNA. Okay? It will actually, as highlighted by the actual arrow in the figure, solid arrow, you're going from here back to this original molecule. That is what, at this point, this RNA polymerase will do. As in, it will dock at this promoter region, this docking site, and it will make RNA. That is what RNA polymerase does. It generates single-stranded RNA using double-stranded DNA. Or rather, double-stranded DNA as a docking site. Provided you give it, the appropriate ingredients. So these are what's required. Pretty much you can think of them as the ingredients required to make the RNA. The NTPs stand for nucleotide triphosphates. And then here at the bottom, the DNTPs, so sort of as mentioned, there are differences between RNA and DNA. DNA typically double-stranded, RNA single-stranded. And so these DNTPs, so DNA stands for deoxyribonucleic acid. And that's why they are using here DNTPs, because they want to make DNA. Okay, they want to make this double-stranded DNA. And so this D, get rid of some of the stuff. This D here in the DNTPs, that just stands for deoxyribonucleotide triphosphates. As in, these are the ingredients to make DNA. That's the deoxy part of deoxyribonucleic acid. But then, when making RNA, using the RNA polymerase, they use NTPs. So RNA stands for ribonucleic acids. And so they are not deoxy, and hence there's no D. They're just using NTPs, nucleotide triphosphates. And so that is pretty much the cycle. As in, to pretty much recap, you start with this molecule, and all it does is, through its activity, it attaches, this small piece that is both RNA and DNA. And then this new piece of DNA, it binds at a specific region, on this new molecule. That's not a good enough story. On this entire new molecule, binding at a specific region. And then the reverse transcriptase, it basically fills in the rest here. Actually, what it is actually doing, and that's why you have this molecule here. But what is actually most important for the last step is this docking site. This double-stranded DNA docking site where the RNA polymerase can come in and regenerate this original molecule, but maybe with some mutation. It's sort of, well, that's how natural selection works. It's chance, because all these enzymes, RNA polymerase, reverse transcriptase, they have error rates. They are not perfect, just as no human is perfect, no enzyme is perfect. And so the same thing holds true at sort of a molecular level. It will not always do everything perfectly, and there's a small amount of error associated with it. So that is pretty much like their overall system. They're going in a circle. Their ultimate goal is to regenerate this, okay? And throughout this entire process, that is circular. This is why it's called, in vitro evolution, because this can all occur in a single tube, and it will just continue going until it runs out of ingredients. Until the limiting factor will largely be the NTPs and the NTPs. Because in this step here at the bottom, to go from here, okay, maybe use a different color. This being filled out. And that's pretty much like the primary difference, or that is the only difference, going from the right to the left. And it's using these to, fill in that gap, okay, to make that now new DNA. So that's why Those are the ingredients that are required. And so, like, this reaction, going in circles over and over, it will happen until it runs out pretty much of these ingredients, the NTPs, and, NTP. And that's how you can pretty much carry out multiple generations in a single tube. So that is their system for in vitro evolution. And so going back to the paper, sort of like how they describe it, words, see if it maybe becomes slightly more understandable, but maybe not. So, yeah, in this system, RNA enzymes are challenged to ligate an oligonucleotide substrate containing the sequence of an RNA polymerase promoter element to their 5' ends. Yeah, they're describing this system, and they're saying in our system, it's designed to basically get to this point at the bottom left, where you have this, as I said, this docking site for the RNA polymerase. The RNA polymerase remakes this original molecule, but maybe with some mistakes. A primer complementary to the 3' end of the enzyme is extended by reverse transcriptase to generate complementary DNA, or cDNA. So going back to this schematic, when they say cDNA, that is that right there at the bottom. That is the complementary cDNA using that reverse transcriptase. That is what they are referring to. So the cDNAs derived from reacted RNAs are then transcribed by RNA polymerase to generate many progeny RNA enzymes. Through repeated rounds of these steps, all carried out within a common reaction mixture, the population expands exponentially. Growth can be continued indefinitely by performing a serial transfer procedure, whereby each transfer provides a fresh supply of reaction materials. And when they say reaction materials, they are referring those ENTPs and those NTP. Those are largely the limiting factors that will stop this from going forever. If they had an unlimited supply, basically, of those DNTPs and those NTPs, it can pretty much continue forever, hypothetically. I'm sure there's other limiting factors, but those are like the large limiting factors. But if they just transfer it, or if they just add in a fresh set of those DNTPs or NTPs, it'll just continue going. Xeron, welcome. Great to see you. Here's what the title: just acceptance of AI or acceptance of something more specific. Oh, is my title wrong? I think, oh, you're probably referring to the last stream I did. Well, I'm not. So yeah, you can definitely watch the VOD either on Twitch or YouTube. But I covered this interesting paper. Find it. Under my AI research that I have a folder for. I covered this paper. Oh, it still has that title? Oh. My bad. Thank you for highlighting that to me. But to answer your question, it was general, uh, rather acceptance of AI in general. Okay, that's super appreciated. I didn't realize. Wrong stream. And yet, I was referring to my stream title multiple times. So understandably sourced. Where was I? Yeah, so that's their system. Pretty much, like I said, circular. It just goes round and round until it runs out of material. And they can supply it with new materials, and it'll just keep going. So that is their evolution system in a nutshell. And that's why they call it continuous in vitro evolution, because again, they can pretty much like get it to go on forever. They really wanted to. Maybe not forever, but like a really long time. Continuous in vitro evolution first was applied to a variant of the class 1 RNA ligase enzyme. So they're describing just, a certain flavor of this molecule here. This is like the RNA ligase. This is just, the enzyme that links two RNA molecules together, basically. And that's why it's called an RNA ligase, because it ligates RNA. We try to be descriptive in our naming conventions. RNA polymerase, it makes RNA using a DNA template. But yeah, which has an unusually fast catalytic rate of greater than 100 per minute. So they're pretty much, so things to keep in mind is like, this schematic here, it's depicting one round. But bear in mind, this is like, in a test tube or like a small tube in solution, and there are, tens of thousands or like millions of copies of this thing in solution. And so, they don't just have like one molecule and just like this one molecule over and over. It's like they have, it's a solution, it's a reaction mixture. So you have, tens of millions, whatever, billions of copies of this. Just keep that in mind, and it's a dynamic system. And what they're talking about here, the catalytic rate, they're referring to pretty much how fast it can ligate the two RNA molecules together. So it's pretty much referring to going from here to here, that's not an arrow, but pretty much like that first step. And it's basically describing how fast it can do that, okay? Since, speed and efficiency matter, and, not all enzymes work at the same rate, or work at the same speed and efficiency. And they're saying here that it has an unusually fast catalytic rate, as in it can carry out this, first step here really fast, basically. And they give it like a specific number. And they say over 100 per minute. Pretty much like, you can pretty much think of it that way, as in it can carry out 100 of these, this first step, in a minute. Whoopsies. So several rounds of stepwise evolution first were carried out to adapt the scale 1 ligase to the promoter-containing substrate and to the reaction conditions required by reverse transcriptase and RNA polymerase. So they're describing past research. So, as mentioned, the RNA ligase, it just it's designed to attach two RNA molecules together. But as mentioned, in this first step of the reaction, yes, you have RNA here, but really, this entire thing, it's both RNA and DNA. So what they're describing is that basically they're talking about CL1, so they're talking about a specific version of these things. And they're saying, well, it wasn't originally able to carry out this reaction, okay? Using this hybrid of both RNA and DNA. Pretty much that was pretty much what their past research was about. It was about, hey, how do we get this CL1 RNA ligase to accept this? Okay, that is what they did. That's why I mentioned at the beginning, they're expanding on past research. So they had to carry out previous evolution, and now they're, using this evolve system to study further evolution. And that's why they pretty much say that they had to do stepwise evolution. First were carried out to adapt the CL1 ligase to the promoter-containing substrate and to the reaction conditions required by reverse transcriptase and RNA polymerase. As in, like, basically, it can do this now, okay? It can participate in this entire cycle now, that's depicted here, but it originally wasn't able to do that. Now that it can, they're going to take advantage of that. The resulting RNA enzymes were then used to initiate continuous evolution performed by serial transfer. After 100 rounds of 1,000-fold growth and dilution, the enzymes had adapted to the conditions of continuous evolution, with a typical individual isolated from the population exhibiting a K-cat of 21 per minute and Km of 1.7 micromolar, blah, blah, blah. So all they're pretty much like saying is they're describing the parameters of like their now evolved enzyme. And they say how many rounds and growth the enzymes had adapted to the conditions of continuous evolution. The typical individual isolated from the population exhibiting, yeah. So they're pretty much saying how fast they are. And this term here, AM, you can think of it as affinity. Yeah. Okay. So going back. So as mentioned, they're working now with this specific CL1 RNA ligase. Like in the original system, this is CL1. And then they say, it can now carry out this, first reaction at a specific speed. Okay, that's what that K-cat number represents. And they're saying, well, you can think of it as, a car going whatever, 70 kilometers per hour. As in, like, this CL1, it can carry out this reaction at this speed. That's what this K-cat number sort of represents. And they think of it as the speed. And then this AM variable that they're referring to, okay, AM, that is, the Michaelis constant. You can essentially think of AM as, affinity. And all it describes is basically how strong these two things interact. Because, as mentioned, the CL1 is an RNA ligase. It attaches RNA molecules together. From this specific reaction, we want to end up here, okay? And again, all it's doing is attaching this small molecule here that contains some RNA. And so to do that, it has to bind to it. It has to interact with it, okay? And when two things interact, there's essentially an attraction, okay? And we can characterize how strong that attraction is. That's pretty much sort of what that KM represents. Of like, how strong is the attraction? I guess to be a little more technical, how strong is the binding between these two things? Because in order for them to be attached together, they have to bind and interact at some point to get to here. And so that is what KM represents. So it's just two parameters where this KCat term is to describe, well, how fast is the reaction? And then this KM is pretty much just trying to describe, well, how strongly do these two interact, okay? And when it comes to KCat, that is pretty much, a linear relationship, as in the higher the number, pretty simple and straightforward. The higher the number, the faster it'll go. Just like speed. Although, not a perfect analogy with speed, but conceptually, it works here just fine. As opposed to KM, and the strength of the interaction between those two things that I just highlighted, that is an inverse relationship. As in, the lower the number, okay, the stronger the interaction. That is what is pretty much important to, I guess, get across. They talk about these. Because, they're going to be talking about evolution. Well, they are talking about evolution. And they're talking about evolution of this CL1 RNA enzyme. Okay, but they need a way...
Welcome to Flavor
Time
Country.
Scroll, thank you so much for the follow. Super appreciated. Happy to have you here. But they need a way to, track it, to characterize, what's going on here. They can't characterize it at a qualitative level. They want to use real, actual numbers that, will change. You're a fellow chemist. Well, everyone is welcome here, as long as they're respectful. Yes, I am a biochemist. Well, biochemist / molecular biologist. And if you're a fellow chemist, welcome. Great to have you. And if you want to share anything about your chemistry background or biochemistry background, by all means. But right now we're going over this paper and basically in vitro evolution. So yeah, they're going to use these parameters to track what's going on, of how our RNA enzymes are evolving. Are they getting better? Are they getting worse? To put it in the context of the actual numbers, is this number increasing? As in, is the speed increasing? Therefore, you can think of it as, is the enzyme getting better, or is it slowing down? And so that is why they're using these parameters. That's why, and these are, standard biochemistry parameters for, describing enzymes. Run-of-the-mill biochemistry that I teach undergrads, or have taught undergrads in the past. So, variants of the CL1 RNA enzyme have been used to initiate additional continuous evolution experiments, which have aimed to discover new catalytic behaviors and to test hypotheses relevant to evolutionary biology. Well, we all have our strengths and weaknesses, myself included. I'm definitely way stronger in biochemistry than, strict chemistry. I guess. Yeah, organic chemistry, I would say not that strong. Or, average, maybe slightly above average at best. I definitely would put, let's say, organic chemistry as a strength of mine. And definitely not nanochemistry. So yeah, for example, CL1 enzymes have been evolved that can tolerate a lower concentration of magnesium, resist attack by an RNA-cleaving DNA enzyme, employ substantially reduced concentration of substrate, or operate at either higher or lower pH. So they're pretty much describing, when they have carried out evolution on this, CL1 RNA enzyme, they have basically got it to adapt to various conditions. Arguably increase the fitness of this CL1 enzyme, where it appears to be more temperature tolerant, I think they mentioned specifically. Other experiments. Operate at either higher or lower pH. Can tolerate a lower concentration of magnesium, and then resist attack by an RNA-cleaving DNA enzyme. So that's actually pretty cool. as in through their in vitro evolution efforts, they were able to confer protection against another enzyme that would destroy it. Okay, so pretty cool. Other experiments involving multiple parallel lineages of continuous evolution have been used to investigate conditions that favor recurrent evolutionary endpoints, starting from the same initial conditions, or that result in extinction through the progressive accumulation of a deleterious mutation. Nothing super critical there. So now they describe pretty much another RNA ligase that they've evolved from past research. So this is why, like I said, I had to go back previous research that was conducted, because this is like, you know, two lines of research that they carried out coming together. They had this CL1 enzyme that they evolved, okay, and then now they have this other one that they call DSL, and that they evolved that as well from past research. And then now, okay, they have the two distinct RNA enzymes. Okay, they describe the first one, CL1. Now they're going to describe this second one, DSL. And now, okay, as we're going to see, hopefully, that, okay, how do these behave when they compete against each other, right? Since, you know, evolution is, you know, a competition, so to speak, you know, survival of the fittest. What they're going to do is they're going to see, like, okay, what, we have this CL1 enzyme, we evolved it independently. We have this other one, DSL, we evolved it independently. Well, now what happens when we put them together in the same tube, and they have to, like, compete against one another, and they have to, you know, go through natural selection against each other. So now they're describing their second RNA enzyme. Recently, a second RNA ligase enzyme, termed DSL, was made to undergo continuous in vitro evolution, and it's, you know, the same, still the same process here I went through overall. And as with the CL1 ligase, it first was necessary to conduct stepwise evolution to adapt the DSL enzyme to the promoter-containing substrate and the conditions required for continuous evolution. The resulting RNA enzymes were then used to initiate evolution by serial transfer, ultimately giving rise to molecules with a kcat of 1.4 per minute and km of 0.14 micromolar. So they're using these same parameters. They described those parameters for that CL1 enzyme, okay? They pretty much said, okay, we have the CL1, and here are, you know, the variables that matter. How fast it is, okay? And, you know, how strong it is when it interacts with the thing that, you know, it's substrate, the thing that it's supposed to bind to. And then now they have this other one, DSL, and they pretty much underwent the same thing. And they're looking at the same parameters, but, you know, they get different numbers. As in this one has a different speed. It has a slower speed, much slower. Like 10-fold, 15-fold slower. The other one had 21 per minute. And then now this DSL has 1.4 per minute. And then the other one had a km of 0.17. And then this one has a km of 0.14. So it's actually interesting, because, so you can think of it as this CL1 that had these parameters. It's faster, okay, but it binds to its substrate weakly, or weaker in comparison to the other one. As in, it binds to this right here, less strong, but it's fast. Pretty much the mirror image for the other one. As in the DSL, it has 1.4, so it's slower, okay? But it has a km, and as mentioned earlier, the km is an inverse relationship, so the lower the number, the stronger, you know, the attraction, so to speak. So in the case, going back to when describing now they're describing DSL. Think of this as DSL. And like, yeah, here, kcat I'm obviously not writing super effectively, but the KCAT, it's slower. But the KM, its affinity to this appears to be higher, which is not what you would expect. And they touch upon that sort of later on to go through further experiments because they get a similar finding later on. But they're pretty much establishing like the baseline. We have these two RNA enzymes that, we treated them independently, CL1 and DSL. So the CL1 and DSL enzymes catalyze the same chemical reaction but have completely independent origins and no sequence relatedness, other than in the template region of the enzyme that binds the oligonucleotide substrate. So, yeah, as mentioned, they evolve them independently. They have independent origin. So, arguably, no evolutionary relationship. They happen to carry out the same thing, but they pretty much like got to the same place through independent, separate routes. Heretofore, continuous in vitro evolution has been used to study the biochemical adaptation of a particular RNA enzyme. So yeah, up until now, they have been carrying out this continuous evolution, and they pretty much carried them out separately. In one case, in one past research paper, they used CL1, and they published that. And then in another research paper, if I remember correctly, they did DSL, they evolved that, and now they're going to, like I said, put them together or have them compete against each other, kind of like see what happens, or and manipulate certain parameters and see what happens or how it affects the outcome. Pretty much carry out natural selection. So now that there are two distinct enzymes capable of undergoing continuous evolution, it should be possible to study their co-adaptation within a common environment. In nature, adaptation and complexity can arise from interactions between different organisms that share a common habitat. Laboratory evolution involving two distinct species of RNA enzymes make it possible to conduct studies in experimental molecular ecology with carefully controlled parameters and detailed knowledge of the evolutionary progression of genotype and phenotype. In the present study, two co-evolving RNA enzymes were presented with a mixture of several potential substrates to provide an opportunity for niche partitioning. Once each enzyme had adapted to a distinct niche, two could persist in a common environment. Okay, yeah. So, what they're doing, as they say, in the present study, two co-evolving RNA enzymes were presented with a mixture of several potential substrates to provide an opportunity for niche partitioning. So, going back to their system, as mentioned before, in past research, it was either they were working with that CL1 or the DSL, but now they're working with both. So, CL1 plus DSL. And now what they're going to do, okay, since, they both kind of go after the same thing, this here, but there is a little bit of, for lack of a better term, promiscuity that they can carry out. As in, this right here, that is, as mentioned, that's DNA and RNA. So you have that small stretch in the black line, that's a small segment of RNA, and then the rest, the open box line, that's DNA. And that is a specific sequence. Okay, that'll be a specific stretch of RNA, DNA, and that matters. That is pretty much what allows either the CL1 or DSL to be selective. It's not just going to bind and interact with any stretch of DNA and RNA going to bind with a specific sequence, or largely specific sequence. There is some promiscuity or room for sort of error or flexibility, but yeah, you can't just introduce any random stretch of DNA or RNA. As in this right here, it can't be just any random sequence of DNA and RNA. There's some amount of specificity. But at the end of the day, the CL1 and the DSL, they go after the same thing, but with some flexibility. And what they're going to do is like they're going to provide it with sort of multiple options, okay? As in they're going to have to compete against one another. You can pretty much think of it as competing for the same food. Although they're not consuming it here, they're generating, this new molecule that, goes round and round through the rest of the reaction. But like, yeah, they're pretty much competing for the same food, so to speak. But it's not a consumption thing. But they're selective in the food that they eat, so to speak. So, yeah, that's why, like I said, they're going to be competing against each other. So once each enzyme had adapted to a distinct niche, the two could persist in a common environment, demonstrating an evolutionarily stable strategy in the context of a laboratory evolution. So, and here in this figure, they basically, they're describing their CL1 and DSL enzymes, and they are showing the substrates that they use. It's like this little patch in my cursor. Here it's would be, oh yeah, context of this one. It's, well, I can't highlight it specifically, but it's pretty much here on the bottom left. And then here, it's at the top left. But it's pretty much the overall same concept. They're just like zooming in on here's what it actually looks like, and here's the actual sequence. That's why you're seeing letters. So, the CL1 and DSL enzymes first were challenged to undergo continuous evolution within a common reaction mixture, employing limited amounts of a single oligonucleotide substrate. This process quickly led to the extinction of one or the other enzyme, depending on which substrate was used. Right, so pretty much, like I said, they got them to compete against one another. They had CL1 and DSL in the same reaction mixture, and they got them to compete for, their food. In this case, a little stretch of DNA and RNA. And then as they described, pretty much only one of them won out, okay? One of them went extinct. As in, one of them won the competition. And that will come down to pretty much those numbers that they gave earlier of Kcat and Km, basically, how fast and how strong it can bind to their substrate here for the stretch of RNA. So basically, when they put them together and they're basically competing for the same food, one of them lives, one of them dies. Essentially a fight to the death. Survival of the fittest. That's why they say this process quickly led to the extinction of one or the other enzyme, depending on which substrate was used. A substrate similar to that used in the evolutionary development of the CL1 enzyme was used more efficiently by that enzyme, and likewise for the DSL enzyme and its preferred substrate. So all they're saying is like, when they provide the preferred food, in this case, make up substrate as food, but again, they're not consuming it, they're just carrying out this reaction. They give the preferred food to CL1, well, no surprise, CL1 wins. But when they do the other, give the preferred food to DSL, DSL wins. Which, That shouldn't really surprise anybody, but it's good that they actually confirmed that. So this is basically just, arguably the simplest, co-evolution experiment. A substrate similar, oh, I already read that. So this advantage could not be overcome by evolution before extinction of the disadvantaged enzyme occurred. By staggering the starting concentration of the two enzymes, it was possible to delay the onset of extinction. Right. So they try a little bit to manipulate the outcome of like, hey, can we maybe, give a boost to one of them? It would be like, so before, oopsies, wanted. So, when they first start the experiments, they start them off at an equal footings, okay? And then what matters on who wins the race is like, which one was given the preferred food. As in, if CL1 got its preferred food, then CL1 wins. DSL got its preferred food, well, then DSL wins. Then what they do is they try to ask the question is like, okay, can we manipulate the outcome? Can we basically give a head start to one of them? And they do that by manipulating the concentration, as in the quantities that are present in the reaction. As in, like, can we give a head start of like, well, we provide 10 units of DSL, just 1 unit of CL1. Will that be enough to get DSL to win, even if we have provided CL1's preferred food? Basically, can we get DSL to win the race? Even though CL1 is faster and we're given its preferred food, if we give it a head start by just putting more of it in the reaction mixture, does that prevent it from going extinct? And if I remember correctly, the answer appears to be no. Or like they can delay it, but at the end of the day, giving it even just like a head start does not stop it from going extinct. So yeah, as they say, it was possible to delay the onset of extinction. So it became clear, however, that additional constraints would be required to prevent runaway growth of the more advantageous species. So yeah, in order to pretty much like prevent basically one of them from like completely winning out and just like demolishing the competition, they basically have to give more of a boost to the other one, or like they have to give a disadvantage to the one that has the natural advantage, or they have to give an artificial disadvantage, one that has the natural advantage. So a serial transfer experiment was initiated, during which a 14-mer antisense oligodeoxynucleotide was used to inhibit the growth of the dominant ligase. Antisense oligos were designed to hybridize to a critical region of each of the two RNA enzymes, thus impairing their catalytic activity. Beginning with a population of 1 picomole each of randomized variants of the CO1 DSL, blah, blah, blah. Yeah. So now they're pretty much, tweaking the conditions to see, how much they can, give an advantage or disadvantage to one or the other. They're doing it in a very specific way. And one of the ways, make sure I read it correctly, transfer initiated during which a 14-mer antisense deoxynucleotide was used to inhibit the growth of the dominant ligase. Antisenses were designed to hybridize. Yeah, okay. One of the ways that they kind of give a disadvantage to one of them. So when they say antisense oligos were designed, so as mentioned, RNA is single-stranded, and it's composed think, or eraser, whatever. RNA is single-stranded. Wanted. And it's composed of, 4 ingredients, the 4, bases. For DNA, you have A, E, C, G. That's for DNA. For RNA, it's pretty much the same. Except you have A, U, C, B, if I remember correctly. But not that it matters all that much. But you pretty much have like 4 bases. And yeah, A binds with E, and binds with B. So RNA is single-stranded, and going back, over here, they zoom in and they show you the actual sequence of, the CL1 and the DSL. And you can hopefully see that, it's a sequence of letters, either A, U, C, or G. And you can follow it from start to finish. Starts up here at the 3, ends at the 5. You have this whole spiral thing, etc., etc. And the small lines between the letters, as in starting at 3, and you end up at the C here, okay? Now, that line between them, that means that they are bonded together. And that's why I mentioned A binds with T and C binds G. And so it's binding to certain portions of itself. That's intentional. Well, hopefully you can kind of see that it has a very specific structure, even though it's single-stranded. Not just like this long strand that's floating around. It actually has, structure to it. That's why they are depicting it in this like very specific way, as in like it looks basically like this. Obviously, it's going to be more dynamic. In reality, that is overall what it looks like. 2-dimensional snapshot. And there are regions, as highlighted by, the solid black lines between the letters, where for some regions you have binding, other regions you don't. And so when they say antisense oligos were designed to hybridize to a critical region of each of the 2 RNA enzymes, thus impairing their catalytic activity, what they're saying here is like they have provided stretches of RNA, okay, that will bind to this, okay, and pretty much inhibit, kind of like destroy some of its activity. Not completely, but just like impair it. You can kind of think of it as, going back to the race example, it would be like giving a weighted vest to like a sprinter. Like, well, we're just going to add, a 20-pound weighted vest to the sprinter. That's essentially what they're doing when they say with the oligos. As in, to give sort of the most simplest example, if the entire sequence was just composed of, U, okay? As in, like, this represents all of, for example, CL1. Well, then what they can do is they can provide, artificially, a stretch of A, and then pretty much, as mentioned, the difference between DNA and RNA is in DNA, A binds with T, RNA, A binds U. And so what will happen is if they artificially provide, this sequence, this stretch of A's, or sort of simplified CL1 that's just a bunch of U's, well then they will actually bind together, and that will like inhibit it. As in, like, you have this extra RNA stuck to it. It doesn't want that. That's not part of it. Like I said, pretty much like giving a weighted vest to a sprinter or a runner, and so that will impair some of its activity. So that is essentially what they are saying here. When they say antisense, as in, it's like the opposite of the original sense. That's sort of the terminology in molecular biology. You have the sense, okay, the actual thing. Going back to my example, the sense would be those stretches of U's. The antisense would be the A, because that is what it will bind to. Then E binds with G, and then A binds with U. And so that's what they mean by antisense. Antisense oligos is like, yeah, the RNA-based nucleotides. And they were artificially made to bind to a certain region on here. And to hybridize to a critical region of each of the two RNA enzymes, thus impairing their catalytic activity. They're pretty much either going to put a weighted vest on CL1 or like a weighted vest on DSL to see how, if we can kind of like level the playing field when we have to compete against one another. Beginning with a population of 1 picomole of each of randomized variants of the CL1 and DSL enzymes, 40. So this is this 1 PMOL. That's just, you could think of it as a small quantity, analogous to like whatever 10 apples. It's just highlighting the specific quantity that they are using. 40 serial transfers of continuous coevolution were carried out, monitoring the concentration of each ligase before and after each transfer. To maintain diversity within the evolving population, error-prone PCR was performed on both enzymes after transfers 10, 20, and 30. Extinction was narrowly avoided during this coevolutionary process by employing the antisense oligos, but it required knowledge by the experimenter of which sequence to target and manipulation of the reaction conditions based on which enzyme required antisense inhibition. Furthermore, the enzymes began to develop resistance to the antisense oligos by acquiring escape mutations that destabilize the relevant base pairing interactions. Prolonged use of this approach would require that the oligos be redesigned to maintain the desired effect. So what they're describing here is, they put on, that weighted vest, but it was a designed weighted vest. It was that stretch of these antisense oligos. What they're saying here is like they had to be deliberate. It couldn't just be a random sequence, because using this one as an example, there are multiple points on the overall structure that you can put the weighted vest on. Okay, you can put it down here at the bottom, or you can put it up here at the top, or somewhere in between. And what they're saying is like, well, it seems to matter where we put the weighted vest, impair the activity of one of them in our attempt to like level the playing field. That's why they say extinction was narrowly avoided during this coevolutionary process by employing the antisense oligo. They say it required knowledge by the experimenter of which sequence to target, and when they say sequence, they talk about like which stretch of like the RNA letters to target. Do we target this stretch, or do you target the stretch up here, or anywhere in between? What they're saying is like it matters. You can't just like randomly target. We actually need to, actually target. So furthermore, the enzymes began to develop resistance to the antisense oligos by acquiring escape mutations that destabilize the relevant base pairing interactions. Prolonged use of this approach would require that the oligos be redesigned to maintain the desired effect. Right. This is, they're pretty much sort of like observing evolution in real time. What they're saying is like, yeah, they can target the weighted vest, but given that, they are carrying out in vitro evolution, they pretty much, it seems like they evolved an ability to resist weighted vest. Because remember, they're using a specific sequence. They're using a specific sequence to target somewhere. Go back to my example. A simplified example. Should maybe. Where, yes, if you have CL1, and let's say, again, going back, it was just a stretch of A's, okay? And yes, if they provided artificially, a stretch of U's, they'll bind, okay? And like it will inhibit some of CL1's activity. But then, since they are doing evolution in vitro, okay, so through multiple rounds of evolution, at some point it looks like, providing the stretch of U's artificially, that is, use evolutionary terms, that is selective pressure, okay? As in, like, since you're adding a weighted vest to CL1, well, you're decreasing, its fitness. Well, then, it's not with any intention, but through a long enough timeline, it's going to, develop a resistance to the weighted vest. And, one of the ways it's going to do that is like, well, if, my stretch of A's is posing a problem because there's this stretch of U's that's binding, well, then it can just like change its sequence. Well, this can just turn into a G. This can also just turn into a G. And G doesn't bind U. It's only an RNA. It's A binds with U, then G binds with C. Like if CL1, change some of its sequence here to just a bunch of G's, well, then this stretch of U's no longer bind, and pretty much, will have pretty much prevented the weighted vest. And the disadvantage or the impairment is gone. But, as they say, prolonged use of this approach would require that the oligos be redesigned to maintain, so, they can, they can overcome what the evolution is doing, but they just, need to pretty much like provide a fresh set of newly designed oligos, or like newly designed RNA that will now bind to this new sequence. As in, to use, going back to my example, this is like the arms race in evolution, where, okay, if we provide this stretch of U's that binds to this stretch of A's on CL1, and then in response, CL1 evolves a bunch of G's, okay, well, then we'll just come in with a stretch of C's, okay? And then, we just like go on forever in the arms race. That's essentially what they are referring to. And so now, onto a separate experiment, a second continuous coevolution experiment was initiated, employing a mixture of 5 different substrates. Each substrate contained a single nucleotide change within the promoter sequence compared with the substrate that was used previously. This change reduced the efficiency of the promoter to 9% to 63% relative to that of the wild-type promoter, and created a single nucleotide mismatch for each combination of enzyme and substrate. Okay. So, let me just make sure I have this correct. Okay, yeah. So, all they're saying here is back to the original figure. They're doing, their coevolution experiment. And now what they're doing is, as mentioned, there's a little bit of specificity in, what CL1 and DSL consume. but there's a little bit of flexibility. And so what they do is they provide, that's not a straight line. I can't draw a straight line all that great. But now they're providing 5 different sets of food. And now they're having them compete. And each of them can sort of like, they each basically have their own preference. Where now, before, they put them together and they have them compete against, one single food, and one single food that they both, enjoy. Now they're together, and now there's sort of a little bit of a buffet. Where now you have more options. Like, hey, what are you going to eat? And they don't have to go after the exact same thing. Whoops. And that's what they're referring to when they say a mixture of 5 different substrates. Kind of think of it like food, but they're not like consuming it and digesting it. They're just carrying out the reaction. And so now it's just, 5 different ones. And all they're saying is like, yeah, when we provide sort of a buffet or a variety of foods, like, yeah, everything still works, but it works with less efficiency. That's all. As in 9 to 63% relative to that of wild type promoter. As in relative to, the actual food that they prefer, actual preference, everything still works, but, just not as good. Continuous coevolution was initiated with, this quantity of the CL1 and this quantity of the DSL enzymes. So now they're playing around with the quantities. They're basically giving now what happens when we give more of CL1, and it's like a 10-to-1 ratio, 10 picomoles versus 1 picomoles. Just think of it as 10 units versus 1 unit. Basically, in this case, CL1 has the advantage if there's more of it, which were transcribed from templates that have been subject to mutagenic PCR. The concentration of each ligase was measured before and after each transfer. And the time between transfers was adjusted periodically to allow the enzymes to exhaust the supply of their corresponding substrates. The concentration of the CL, blah, 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 staggered. Yes. Okay, nothing super critical here. The initial reaction mixture contained 1 micromole of each of the 5 substrates. Assays were conducted before the start of the experiment and periodically thereafter to measure the ability of each enzyme to amplify in the presence of 1 micromole of each substrate, tested individually. Both enzymes showed a strong tendency to adapt utilization of S5, and accordingly, the concentration of S5 is reduced to 1 micromolar or eliminated completely for individuals. Okay, yeah. So here, they pretty much carry out roughly this. It's like you have the presence of both CL1 cell, and they have their 5 varieties of foods / substrates. And then now, what they're saying is, like, in the presence of the 5 different ones, they evolve a preference for like one specific one. As in, like, in the buffet of the food, they both seem to evolve a preference for one particular food or substrate language. And they say that or they highlight that by showing how this S5, so that's one of the foods, the buffet, and they say that they start off with, one specific quantity, as in 1 micromolar, okay, and then this S5 was reduced to 0.1 micromolar, so it went down, it decreased. That's to say that, a significant portion was consumed. That is their evidence. So that, okay, presence of both, like this S5 substrate, they seem to consume a lot of it. So they're evolving a preference for that one. After 50 transfers of continuous coevolution, with an overall amplification of 10 to 100 fold, individual CL1 and DSL molecules were cloned for the population and sequenced. A typical clone of each enzyme was chosen for more detailed analysis. The evolved CL1 enzyme contained 5 mutations relative to the starting enzyme, whereas the evolved DSL enzyme contained 11 mutations. So here, they get down to the nitty-gritty of like, okay, something is changing about the CL1 and DSL enzymes. So what exactly is changing? Say what? Pretty much referring to the sequence of letters. Oh, this EUUAUG. I think they highlight, yeah, they do highlight the mutations. Yeah, filled circles highlight mutations present in typical clones, isolated after 50 transfers of coevolution with 5 substrates. Yeah. So, the filled circles, it's like this right here is a filled. So they're saying like, oh, there's a mutation here. When they have coevolution, when both of them are, competing against one another, and they're presented with, that buffet of, 5 different foods, they both evolve, pretty much prefer one specific food, but, their pathway to get there appears to be different. And they highlight, the specific pathway. As in, CL1 had 5 mutations, DSL contained 11 mutations. Basically, more mutations occurred in DSL. These mutations included compensatory changes within the portion of the enzyme that binds the substrate. A13G for CL1 and U22A for DSL. So all they're highlighting here is like, yeah, an A at this specific position, the 13th position, was changed to a G, and then this U at the 22nd position was changed to an A DSL. And these appear to be like critical, important mutation of like, hey, this is what allows them to prefer this specific food when presented with the buffet, the 5 different options. Providing perfect complementarity with substrates S4 and S5 respectively. Amplification profiles were obtained for each of the evolved enzymes, demonstrating mutually exclusive use of S4 by CL1 and of S5 by CL. So let me see. Oh, okay, yeah. So, at some point, there's a little bit of divergence. Okay, like in some experiments, when they have them compete against one another, and they provide them with the 5 different options of food, like, yeah, they both seem to gravitate towards that one specific food. In this case, starting off, both enzymes showed a strong tendency to adapt to utilization of S5. Okay, so this particular one out of the set of 5 that they present. But then, upon further coevolution, like I said, carrying it out, this can pretty much like go on forever, just provided that they give more fresh ingredients. But then at some point, there's a little bit of divergence. Okay, evolution in a test tube in vitro. And so, they don't necessarily, there's an advantage of, like, pretty much choosing a different food, right? If you have the option for something else, you don't want to both be consuming, or rather both be competing for the same resource. And so that's what they highlight here, as in at some point, okay, there's divergence. Where, let's see...
show me the money.
Oh, discount Seth Rogen. Thank you so much for the sub. Super appreciated. Super kind of you. And thank you so much for passing by. And I hope you're doing well. And yeah, 5 months. That long time. That's super generous. And so pretty much at some point, one of them goes after S4 and the other one sticks with S5. As in, like, hey, oh, thank you so much for the lurk. Super appreciate it. And again, can't thank you enough for this. And so, like, yeah, since they're competing for the same food, like at some point on a long enough timeline, after many generations of evolution, one of the enzymes, the RNA enzyme, says, hey, I'm gonna go consume something else. There's no point in both of us competing for the same thing. And so it's like, yeah, demonstrating mutually exclusive use of S4 by CL1 and of S5 by DSL. So yeah, they start off, they go after the same thing, and then at some point, they diverge. You know, one of them gives up, saying, hey, I'll go somewhere else and eat something else. Yeah, exactly. Oh, this is the arms race. This is one way of, you know, generating peace. So the exponential growth rate of DSL appreciably exceeded that of CL1 when each enzyme was allowed to operate in the presence of its preferred substrate. After 30 minutes incubation, the DSL enzyme exhibited 150-fold amplification, whereas the CL1 enzyme exhibited only 13-fold amplification. In addition, DSL achieved a maximum extent of growth that was approximately 3-fold greater than that of CL1. So, you know, there's a little bit of a trade-off, where, yeah, in this cycle, okay, what they're saying here, basically, one, it can, its performance, okay, it was 13-fold amplification after 30 minutes. So, yeah, it's just going through this cycle, you know, over and over, provided, you know, enough of the ingredients, and time-based, you know, they pretty much, you know, they put them in the test tube, and, you know, they just say go, and they start the timer, and then after 30 minutes, you know, they stop, and they assess, like, okay, what happened? What's the outcome? What's the damage? And then CL1, you know, basically, got 13. That is not, but like, yeah, that's pretty much its outcome. 13-fold. And that's after 30 minutes. But then DSL, okay, pretty much performed, you know, over 10 times better. DSL, 150-fold. Because remember, you know, this is a cycle, and the ultimate end goal, you know, as they go round and round, you know, they're just like generating this over and over. That is what they're referring to when they say RNA progeny. Like, that is not how you spell progeny. Yeah, progeny as in, like, you know, their offspring in the RNA enzyme world, as in, like, this is their offspring. So it's like, well, even though this is the starting point, but this is like, given the conditions that, you know, they put them in, the evolution, this is self-replicating, okay? That's what makes this so powerful. Like, it's generating itself, but this is still, you know, the progeny is itself. That's what's, you know, kind of like funny about it. And that's what's so elegant about it, I would say. So what they're saying is, and that's why they say it's an amplification of, like, well, how much, even though this is the starting point, you know, how much of this are you making? And yeah, so for DSL, it's 100-fold. Performed much better than CL1, as in it amplified itself 150-fold, given the starting point, as opposed to CL1, just 13-fold. So basically, DSL had much greater offspring / progeny in comparison to after 30 minutes. And in addition, DSL achieved a maximum extent of growth that was approximately threefold greater than that of C. Yeah. So overall, looks like DSL, won the race, pretty handily, over tenfold greater. The compensatory mutations alone were installed in the CL1 and DSL enzymes. For CL1, the matched pairing between the enzyme and substrate resulted in growth characteristics similar to those of the fully evolved enzyme, suggesting that the other 4 mutations in the evolved enzyme are near neutral with regard to fitness. For DSL, in contrast, the compensatory mutation alone did not provide the full growth rate of the evolved enzyme. DSL is a younger enzyme compared with CL1, having been subject to many fewer rounds of in vitro evolution, and thus is more likely to acquire mutations that enhance its fitness within the context of a continuous in vitro evolution. But what they're saying here is pretty much like commenting on their path that each of the enzymes, the RNA enzymes, undertook, and sort of like explaining like the discrepancy, so to speak. And as they sort of highlight here, DSL is a younger enzyme, and that is likely the reason why it was able to incorporate, if I remember correctly, more mutations. Yeah, as highlight, DSL enzyme contained 11 mutations, as opposed to the 5 L1. And so what they're saying here is like, given the fact that DSL is younger, as in hasn't undergone as many rounds of evolution in comparison to 1, then it can pretty much, its evolutionary window is larger. That's why it can have more mutations. And that's likely their explanation as to why DSL essentially won the arms race. That's why I was able to produce way more progeny. And they're sort of commenting on sort of like the types of mutations that occurred, where some of them appeared to be near neutral, as in it didn't seem like, going back to the mutations, and remember the closed circles is here at the bottom. U, that's an example of a mutation. And what they're saying here is like, looks like some of it for DL1. Not that it matters much, but basically for some of the mutations in either CL1 or DSL, the mutations appeared inconsequential, as in not critical to make it better or worse. It's just like, this is a change that occurred. Because like arguably this U here, that's a mutation, the activity part, where all the business is, appears to be not appears, it is occurring up here. Like this arrow here, this is pretty much like that RNA ligase reaction. Pretty much saying, hey, we're going to take this stretch of RNA, and we're going to attach it to myself. So this is like the business end. And so to have this mutation here, this is probably like more in the near neutral mutation. It's like, well, why is this mutation occurring? Like this does not appear to be a critical that is like enhancing or decreasing the fitness of the enzyme. So it is possible to balance the disparate growth rates of the evolved CL1 and DSL enzymes by adjusting the concentrations of their respective substrate. The CL1 enzyme operating in the presence of 1 micromolar S4 has a nearly identical rate and maximum extent of growth compared with the DSL enzyme operating in the presence of 0.02 micromolar of S5. Right. What they're saying is, as mentioned, when they put them together and they present them with, the buffet on like a long enough time scale, CL1 prefers this part of the buffet and DSL prefers like this other part of the buffet. And now what they're saying here is, well, they can sort of manipulate the outcome by adjusting the conditions, where if CL1 prefers the S4 food, okay, and DSL prefers the S5 food. Okay, they can level the playing field by just manipulating how much of the preferred food they give it. As in, 1 is bigger than 0.02. Okay, and that's why they're saying the CL1 enzyme operating in the presence of basically more of its preferred food has a nearly identical rate and maximum extent of growth compared with the DSL enzyme operating in the presence of 0.02 micromolar of S5. Remember, the DSL basically won the race. Now they're pretty much like saying, well, can we level the playing field? Well, they can by adjusting, how much of the preferred food that they give it. Where in the context of DSL, DSL is the winner and it prefers S5. Well, if we just give it less S5, just like the equivalent of, giving a head start CL1. Give it a head start by providing more of its preferred food in S4. And that's how they're able to level the playing field, so to speak. And as they say, under these conditions, it is possible to carry out the sustained coevolution of CL1 and DSL within a common reaction mixture over the course of a serial transfer experiment. So they're carrying out more evolution, and now that they know, okay, that they've gotten to the point in their in vitro evolution experiments that, okay, CL1 prefers S4, DSL prefers S5. Okay, so they have both evolved their own preferred food, and that's why, going back to pretty much the title of the paper, you have niche partitioning. And now they're going to see what happens. They're going to extend it even further, I believe, of okay, now that they're both occupying their own little niche, what happens now? They just let things play out. So in the absence of further evolutionary change, this behavior could be maintained indefinitely, demonstrating occupancy of distinct niches by the two different RNA enzymes. So they're pretty much saying, well, this state of affairs presumably could just be maintained if there's no other selective pressure placed on the system. Where it's okay, DSL prefers this, CL1 prefers this, and we're just going to mind our own business. As in, if we just keep the status quo, then no need to change. And they're just going to occupy and stay in their own distinct niche. Did I? Oh, yes. So the catalytic activity of the evolved C1 with the preferred substrates was examined under the conditions of continuous evolution. So now, they're going to actually characterize, the parameters, how fast they are of okay, we've subjected DL1, DSL evolution. And, many generations, and, they have their own now unique niches of their preferred foods. Now, what is their actual performance? Really trying to actually characterize them. So that's why it's going back to the numbers. They want to actually quantify it. They don't want to just say well, this one just performed better. It's well, how much better? Can we put a number to it? So CL1 demonstrated a KCAT of 290 per minute. So this is the speed of the enzyme, RNA enzyme. And KM of 17. Pretty much how strongly you can think of it as, how strongly it's attracted to its food. Or I guess you can also kind of think of it as how strongly it prefers its food. And again, it's an inverse relationship when it comes to AM. The lower the number, basically, the stronger the attraction, so to speak. So this is how CL1 it ends up performing. This catalytic rate is among the fastest ever measured for an RNA enzyme. So like at this 290, what they're saying is like, oh, this is super fast. Like basically, when it comes to how fast it is, we've really significantly like increased its fitness in this specific parameter. Because remember, I believe we started off at like, you know, 1, some double-digit number. Yeah, 21. For both of them, they start off as like 21 versus, yeah, 1.4. Now we're ending up at 290, okay? So like pretty much 100-fold, 100 times better. In contrast, DSL exhibited a KCAT of 0.17 and a KM of 0.4. What's interesting, and they're gonna comment on it pretty much in the next paragraph. But like, so remember, I believe DSL is the one that's like the overall winner, at least in one of the experiments. After 30 minutes incubation, DSL 150-fold amplification, DL1 just 13. Oh, going by this metric, you know, DSL's like really the clear winner here. But then, interestingly enough, DSL, it has a much smaller number when it comes to its speed. You know, the speed of CL1 is 290, you know, per minute. And then DSL, it's, you know, 0.17 per minute. It's like way less, over 100-fold, 100 times less than the speed of CL1. But then if you compare their KMs, actually like most, at least I think, but you know, I'm a biochemist. These are like the parameters that I'm used to working with. Yeah, so for CL1, got 17, and then DSL got 0.24. But what's interesting is like CL1 is like faster, okay? It's faster than DSL. But when it comes to like, you know, how strongly it's attracted to its food, or, you know, its substrate, their terminology, DSL is like, yeah, is way better in that regard. As in, the picture is CL1 is fast, but not really all that attracted to its food. As opposed to DSL, basically the mirror image. It's slow, but like it's strongly attracted to its food. It's like, oh, it really wants this. It's going to bind tightly. You know, it's going to move towards its food really fast, but it's going to consume it slowly. As opposed to, again, CL1, not that strongly. Not going to move towards its food really quickly, but once it gets to its food, it's going to consume it really quickly. So not molecular thermodynamics. This is enzyme. No, it's not weird. I don't think it's weird to ask. I mean, like, at the end of the day, you know, if you zoom in close enough, everything is whatever molecular thermodynamics, I guess. But no, this is really like enzyme kinetics, or standard enzyme kinetics. This KM, this is the Michaelis-Menten constant. Yeah. In biochemistry, Michaelis-Menten kinetics. So you have this Km. Yeah, when the value of the Michaelis constant, Km. So that is what it is. Definitely not weird to ask. That's what they're referring to, and that's why, like, we use it in biochemistry, because it has to do with enzyme kinetics. Usually, well, generally speaking, we're using it in the context of protein. But here, it's the overall same principle, but we just happen to be RNA. But the principles are virtually all the same. They're both acting as catalysts. So, yeah. That's pretty much where they end up, and it's an interesting sort of like outcome that is kind of unexpected, and I think they're going to highlight that. So yeah, thus, the two enzymes exhibit markedly different kinetic parameters, with a 100-fold difference in observed rates, as in one is way faster than the other, in the presence of 1 micromolar of their respective substrates, yet both enzymes are viable within the continuous evolution system. Right, so in terms of like performance, okay, they perform dramatically different, but yet they're basically able to coexist in their system. So interestingly, the slower DSL enzyme amplified more efficiently in the continuous evolution mixture. That's pretty much why I highlighted here, where it's like DSL, like in this context, like it won the race. But then when you look at, objective parameters of how it performs, it's like, it doesn't seem to perform all that great. Where? Oh, yes. Yeah, the slower DSL enzyme amplified more efficiently in the continuous evolution mixture, warranting investigation into other factors that might affect amplification rate. The rates of reverse transcription of the two ligated enzymes were measured and found to be 0.2 and 0.3 per minute for CL1 and DSL respectively. However, the rates of forward transcription starting from cDNA were more disparate, the DSL exhibiting a 2.2-fold faster rate of transcription over a range of not super critical. Synthetic DNA templates were prepared to validate the observed difference in transcription rates. Not super critical. But yeah, it's kind of like a little bit of a paradox of like DSL is a winner, but then, I guess to use a car analogy, when we look under the hood, it's like there's doesn't appear to be anything special here, but it's like clearly winning the race and winning the race by a mile. And they try to tease out, basically, in this last description here, of like, eDNA and reverse transcription, they're basically going back, like, okay, where in these steps, clearly this is a multi-step process. They're basically trying to assess, like, hey, where in this process does DSL have some sort of advantage that we can kind of like, well, is it just like super fast here, or super fast here, or super fast here? Not that it's super critical. Well, at least for the purposes of understanding the paper, not super critical. I mean, well, yeah, fair enough. Good analogy. I take your point. That's where it's, what it is evolution just offspring, and just, more offspring is, better evolved? I don't know if evolution can only be boiled down to that, but that is, kind of like the common sentiment. I think most biologists would accept that explanation. So in nature, the competition for limited resources is a critical driving force for adaptation and evolutionary innovation, both among individuals of a given species and between species that occupy the same environment. Continuous in vitro evolution has proven to be a powerful method for evolving RNA molecules in the laboratory, enabling the selective amplification of individuals from a heterogeneous population over the course of hundreds of generations of sustained. Right, so, they're studying evolution, and at the end of the day, they at some point want it to be applicable to humans or have some sort of human application. Not that all lines of research need to apply to humans. I do foundational research, and foundational research is super critical. But, when it comes to how real world science operates, when it comes to funding and grants, what we seem to care about, or what gets funded, is like, hey, how does this apply to humans? And even though they're not talking about humans here, but they're talking about, evolution. Evolution applies to humans. And they're pretty much like, I would say, demonstrating the power of their system. Of like, this is an enclosed system. It's, basically self-sustaining. Yeah, you still need to add like fresh ingredients to get it to keep going because it does run out eventually, but it is largely self-sustaining, and you can pretty much like go through multiple rounds, like generation, as they say, hundreds of generations. Oh, and Rafa is God. Welcome. Hope you're doing well. Always great to see you. If resources are not limited, that's an excellent question. Well, they sort of highlight that here, and well, I highlighted it also. In the absence of further evolutionary change, this behavior could be maintained indefinitely. Demonstrate. So, yeah, use the example of the paper. Whatever. CL1 prefers S4, and DSL prefers S5. So CL1 prefers this one food, and DSL prefers this other food. If they just leave them be, and there's an infinite supply of basically resources, and there's basically no selective pressure, and it's highly unlikely that any sort of evolution would occur. So that's a great question. Thank you so much. And yeah, that is, I think, important to point out, because you need that selective pressure. Valid criticism. They obviously can't say for sure, but I think there are good foundational reasons to say that. Is it possible to say West? So it depends on what you mean exactly. If you want to expand, so because I've sort of heard this sentiment before a little bit, in terms of modern medicine, is providing life-saving healthcare to people that would have died 50 whatever 20 years ago, or whatever 100 years ago. Is that going to slow evolution? I'm not saying that you're saying that. I'm just sort of something that I've heard. But basically, there is actually research, I believe, to back up the claim that I'm making, that I'll try and dig up and maybe cover on a future stream. But I remember coming across multiple papers. But yeah, there's every reason to believe that humans are still evolving. Now, whether it's slowing down or speeding up, I can't say one way or the other. But I don't haven't done any deep dive. But I see no reason to think why human evolution has either sped up or slowed down. But a totally valid question. And yeah, if you want to sort of expand upon what you mean by slowing human evolution, I'm all eyes and ears. But yeah, I think we just evolve differently. So wrapping up, we're in the discussion. Yeah. Starting here. So yeah, pretty much the overall advantage of the system is they're able to go through hundreds of rounds of generations of sustained growth. They're doing kind of a little bit of a mini simulation. It's a huge stretch, but we can't study human evolution, at least not across hundreds of generations. That would take thousands of years, right? But they're doing that in a test tube, in a matter of whatever, hours or days, right? That is the power of their system. This method had only been applied to one class of RNA enzymes at a time. In the present study, 2 unrelated catalytic species were made to undergo continuous evolution within a common environment, competing for limited amounts of substrate. The competitive exclusion principle states that 2 species that compete for the exact same resource within the same environment cannot stably coexist. Such conflicts often are resolved in biological ecosystems by adaptation of the competing species to distinct ecological niches. In the context of continuous evolution, when mixed populations of CL1 and DSL enzymes were challenged to compete for the same substrate, i.e., food, they're going after the same thing, extinction of one species or the other could not be avoided. Right, so going back to here, when they set it up that you have, DL1 and DSL, they're put, on a level playing field. They're pretty much at the same starting position, and they're competing for the same thing. One of them goes extinct, just like, well, it's a winner take all, eventually. And then, as we just went through, once they start manipulating conditions of like presenting more foods or more options, then different outcomes happen. As they highlight. However, when the two enzymes were presented with 5 potential substrates, each enzyme adapted to use a different substrate, i.e. food, demonstrating what is termed ecological character displacement. So it's like, yeah, when they're competing for the same thing, winner takes all. When they present multiple options for winning, okay, then different outcomes. Once differentiated in this way, the two enzymes were capable of sustained coevolution, which in principle could be continued indefinitely. Further evolutionary change might lead to extinction of one species if the other species usurped its substrate before an effective counter-strategy could be evolved. Barring such events, however, the current situation represents a stable strategy for coexistence. Like if our food needs are being more easily met, do we lose our hunter-gatherer instincts? I guess you could say we have devolved in some ways like that. If that is so that's a fair question. Well, I guess it's there's a larger question of do we even have hunter-gatherer instincts? I think I'm not sure the evidence is clear per se that we have instincts, or I think it's hard to define instinct. But I understand the overall. I'll take it as a given. Like we have some sort of default operating system form of instincts. And it's like, yeah, is providing food. Oh, that might be just kind of like, wiping away that default operating system, i.e. instincts. Possible, but I would say even then, even if I grant it that we have instincts, yeah, the food thing, I don't think the abundance of food is like enough selective pressure, at least not in the short term. I guess the best example that I can use is like our appendix, I think, where even though there's a little bit of a debate on whether the appendix is actually useless, and that there appears to be maybe some uses for our appendix, but regardless, I think generally speaking, the appendix poses more problems for humans than not, in the form of like appendicitis or inflammation of the appendix needing to be removed. Decently common. And it's like, at least so far, maybe whatever 1,000, 10,000 years down the road, will have evolved no longer needing an appendix. But as of right now, I think it's fair to say we don't absolutely need our appendix, at least for survival. That's clear. There's some evidence to show that it might have some uses for the microbiome, etc., and repopulating the microbiome. But I think that's still like to be determined. But yeah, at like a fundamental level, clearly we don't need our appendix, and yet still have one, because like as of right now, well, there's even arguably some selective pressure to evolve not needing or not having an appendix, because I think overall it poses more problems, arguably. But as opposed to like instincts, like unless they actually pose a problem, assuming we have them, like I don't see how food would result in our instincts going away. It's just like, you know, they'll be maintained unless there's like good reasons for us to not have them anymore, or there's like some selective pressure for us to like eliminate our instinct. But again, this is all just me speculating. For all thought experiments. That's pretty much how I would understand it. But yeah, good question. Totally valid question. The niche space provided by the 5 substrates is well defined in molecular terms. These substrates differ by 1 or 2 nucleotides within the region of 8 nucleotides that is bound by the RNA enzyme. So it's like, you know, yeah, they can pretty much see at a molecular level like what the food that they're consuming looks like. As in like DSL likes this, and this is what it looks like. I think they even actually, yeah, going back to the figure, it's yeah, right here. It's in the description, where shown with substrates S4 and S5 respectively. So yeah, CL1 likes S4 and DSL likes S5. And so yeah, case of, yeah, CL1, S4 is here. Well, can't really highlight it, but it's, you know, at the bottom left. That stretch there at the bottom left starts with C, E, A, and then like, yeah, the ligation, okay? Enzyme activity occurs here at the top with the arrow. And then here for DSL, this is the actual S5, yeah, S5 substrate. You can actually see the sequence of like DSL, overall molecule, it prefers this stretch here. And then for CL1, prefers that stretch there, the bottom left of it, starting with CTAA. And so we can actually see, like, you know, what it prefers. Not just all theoretical. We can see, oh, CL1 likes this, and DSL likes that. And as they say, you know, there's not a huge difference between the two. As in S4 and S5, they differ by 1 or 2 nucleotides. As in, yeah. Right. So if you actually look at the sequence, like right here in the letters, yeah, it starts off CTAA, and so does the other one, AA. So like pretty much, it's just like small changes. The overall sequence is the same. It's just like really small changes that result in that niche partitioning. Is devolving the wrong term to use, or is it always just called yeah, I don't think devolving is a thing, because, you know, understandably, but I think it's mistaken, and I think other evolutionary experts would agree that there's no such thing as like devolving, because it hints at some sort of like hierarchy of like, oh, humans are more evolved than bacteria. Like at most, you know, I think you could just talk about evolutionary history and talk about generations of evolution. And if anything, you know, bacteria are more evolved than humans, because like they've been around longer and like they've been subjected to evolution longer than humans. But I think overall, yeah, devolving, not really a term. It's just like evolution, because you adapt to your environment, and like there's no better or worse environment. It's just like what allows you to survive. It's like the conditions are the conditions. It's not when something becomes extinct. Is it a result of it not evolving or a result of other things evolving? That is an excellent question. For I think depends on the context. Where, yeah, I don't think it can be boiled down to one or the other. Because I think either or can occur, and also both can occur at the same time. Of like something can, you know, evolve faster, or something can, you know, not evolve. Well, that they sort of cover that a little bit. Yeah, I think it can go. You know, using this example, going back to just the L1 and DSL, like how do you frame it? Like when they're both put at the same starting line on equal footing, okay, and they're both going after the same thing, like this one thing. That is their finding, right? When put both there, the same starting point, and we just say go and go after the same thing, one of them wins out. One of them does go extinct. So do we frame it as, you know, let's say CL1 is the one that survives? Is that because it evolved faster? Or is it because, you know, DSL evolved slow? Or like did not keep up? I don't know that one or the other. I think they're both equally valid, I think. Hard to say. But in the context of like a species or like an animal going extinct, yeah, I think both of like both conditions of like let's say an invasive species, and like an invasive species, you know, eating the food of another species, and like that other species can't keep up with the invasive species. And so I can sort of see it as like, well, one evolving faster, evolving slower. So yeah, that's a complicated question that you'd probably need an actual evolution expert. Yeah, straight enzyme. Which, you know, where nothing changes or nothing happens, you know, I think that's also human hell too. Do you think that is something we can look at in modern humans in terms of cancer and how some people seem to be less likely to get it, or is cancer considered unnatural? No. We are looking at it. So interestingly enough, we know that there are some populations of people that they appear to be protected from some forms of cancer. But they also seem to be at higher risk of other forms of cancer. And trisomy 21, also known as Down syndrome, appears to confer some protection against some cancers. I believe they're at higher risk for other ones. But yeah, 10 reasons why people with Down syndrome are protected from the development of most solid tumors. So, and like, I mean, cancer is natural, so to speak. Not saying it's good, but you know, pretty much, yeah, cancer is almost an inevitability. In whatever, in animals, given how we operate and, you know, replicate. But yeah, going back to cancer protection, there are certainly people, like individuals with Down syndrome slash trisomy 21, they appear to have protection from certain cancers, and we're trying to understand why that is and what the actual mechanisms are. And so assuming we do get an understanding of why, I have real doubt that that would lead to some sort of therapy or understanding cancer way better. Ooh, I mean, if you have, I mean, I can totally believe that. I'm curious, if you happen to have any sort of like link or research that highlights that. But you're not obligated. Where was? Yeah, so like, so yeah, they evolved to, consume their specific food, but even then, the food that they're consuming basically doesn't appear to be all that different. The fitness landscape for each of the two co-evolving enzymes reflects this niche space and potential interactions with the opposing enzyme. In the present study, those interactions were entirely competitive, but other types of interactions, such as commensalism or predation, might have emerged. The enzymes compete during the exponential growth phase for the finite supply of substrates / food, and as they reach the carrying capacity of the reaction mixture, compete for other resources, such as the primer for cDNA synthesis and the NTPs required for transcription. These processes give rise to complex growth dynamics, which nonetheless could be described in precise molecular terms if one measured the detailed kinetic properties of components of the continuous evolution. Right, so all they're saying is, going back to, their simplest of experiments, sort of. But assuming, CL1, and DSL, when they're put together, and, competing for, roughly the same thing, all they're saying is, like, yeah, they compete, but, like, once this is allowed to proceed, they're competing against the same food, they basically start competing for other things. They start competing for this, start competing for this. They basically start competing for other things along the stepwise process. And they're saying, like, there's multiple levels where evolution and competition can take place. It's largely driven by here, but like once this runs out, okay, and assuming both are still around and they both haven't gotten extinct, they start competing for other things along the process, or in this case, along the evolutionary landscape. With your inorganic professor said, do not have research. Yeah, fair enough, but I can totally believe that. And also, given what I know of the safety practices of chemists and what they deal with, totally believe that lab chemist, a quarter of lab chemists. So, according to the RK selection theory, a species will tend toward either of two general evolutionary outcomes when forced to adapt to selection pressures, where R refers to the reproduction and rate, and K refers to the carrying capacity of the local environment. Although actual cases in biology usually are more complicated, R-selected species are exemplified by the rapid production of numerous offspring that are better suited to a fluctuating environment, whereas K-selected species have lower fecundity and more fully exploit the maximum carrying capacity of a stable environment. Right, so they're basically talking about different evolutionary strategies. One is like, carpet bombing with a bunch of offspring, so to speak. And the other one is like, lower fecundity, but, they exploit the environment more. In broad terms, the evolved CL1 enzyme is a K strategist that generates fewer copies, the offspring, per round of replication compared with DSL, but more effectively utilizes the available substrate because of its very rapid rate of catalysis. So this goes back to kind of this here, where DSL appeared to be like a winner in terms of like, well, generating way more progeny or offspring, so to speak, like offspring of itself, self-replicating. And then, but what they're saying is, CL1 sort of was able to overcome this, or its strategy is basically doesn't generate as much progeny, but it's able to like go after its food with super speed. So it's like it makes less of itself, less copies of itself, but it goes after its food. Like a rabid dog. As opposed to DSL, it makes a lot of copies of itself, but doesn't really go after its food. All that rapidly. And Elwa Links, welcome. Thank you so much for passing by. Hope you're doing well. Great to see you. And that is exemplified by the kinetic parameters, this KCAT and KM. That's pretty much speaking to the strategies. Oh, this R versus K evolutionary strategy. Where it's like, it's either a lot of progeny or you're able to exploit your environment better. So almost every CL1 molecule has reacted before it is reverse transcribed and as a result inactivated. The evolved DSL enzyme, in contrast, is an R strategist that generates 2.5-fold more copies per round of replication compared with CL1, but because of its slower catalytic rate, it is more likely to be reverse transcribed. Both strategies are viable and at steady state result in a higher fraction of the CL1 molecules being in the reacted state compared with the DSL molecules. Right, so you're saying here, it's like, the DSL, its evolutionary strategy is like mirror image of CL1, where CL1 is a K strategy that generates fewer copies, but...
Show me the money.
Oh, wow. Thank you so much for the sub. Super generous and super grateful. Yeah, I can't thank you enough. That's super appreciated. And happy to have you here. And so the evolved DSL, its strategy, I'm almost like distracted by the generosity. But yeah, thanks. I don't really know what to say. Where was I? Yeah. So, the CL1, going back to like the diagram. Basically, as they allude to here, when they talk about DSL, is more likely to be reverse transcribed. They're actually talking about this step. So remember, we have CL1 and DSL. Okay, and like, yeah, in the beginning, going after, the same thing, the same food, but as, clearly highlighted here in the figure, you have multiple steps, okay, in their life cycle. Pretty much like refer to this as life cycle, but they're generating themselves. And it's like, yeah, one of them, it has the advantage here in like the food portion, the substrate portion, okay? But as they say, is more likely to be reverse transcribed. So evolved DSL is more likely to be reverse transcribed. They're referring to, this step here at the bottom. So it's kind of like, CL1, it might go after its food, super fast. It might be efficient in this regard, but it seems like DSL, it's less efficient going after its food, but it seems like in this step here, that's where it has its advantage. That's where it seems, that's like it carved out its niche. It's like, okay, you can be fast here in this step, going after, the food / substrate, but it just like compensated by pretty much like being good at this step here. So just like different evolutionary strategy. And as they say, both strategies are viable. And at steady state, result in a higher fraction of the CO1 molecules being in the reacted state compared with the DS. Oh, cool. Oh, well, that's super cool. And welcome from Greece. Or well, I guess I don't know if you're Greek from Greece, but regardless, that's cool. The evolution of niche partitioning for a mixed population of two different catalytic RNAs broadens the scope of complex behaviors that have been realized in a molecular. The disparate kinetic properties of the CL1 and DSL enzymes demonstrate that there is a wider range of adaptive solutions within the continuous evolution system that had previously been thought. The L1 enzyme, with its extraordinary catalytic rate of 290 per minute, so it's like, yeah, it goes after its food super fast, or rather, I should say, once it gets its food, it consumes it super fast. It outruns reverse transcription with an observed rate of blah blah, so that nearly every enzyme molecule is able to give rise to progeny. The DSL enzyme, with a catalytic rate of only, and it's way lower, so 290 versus 0.17, like over 100 times less, or pretty much 100 times slower, over 100 times slower, reacts slower than the observed rate of reverse transcription, but compensates for the reduced number of viable parents by the production of a higher number of progeny molecules per parent. By this enhanced transcription rate, cognate substrate for the evolved DSL enzyme encodes a promoter that is threefold weaker than that encoded by the substrate for the evolved CO. Not a super. This observation suggests that the enhanced transcriptional efficiency of the DSL enzyme derives from the sequence of the transcript itself, as has been observed for. So, wrapping up your overall summary. A long-standing goal of laboratory evolution has been to model complex population behaviors within a molecular system. Isothermal RNA-based amplification methods have been used to evolve non-catalytic molecules that mimic both predation and cooperation behaviors, and they essentially simulated that in this research, right? Maybe not predation per se, but they got one to go extinct. Right? Again, going back to here, once they put them competing against one another on, starting at the same starting point, one of them went extinct. But they were able to level the playing field when they, provide that buffet of, 5 different substrates. Then they both kind of like go their separate evolutionary ways. And, one of them goes after, S4, and one of them goes after S5. Oops, sorry. And those are, I don't know if those are cooperation behaviors. Those are kind of like, well, I guess, coexisting. However, the absence of activity-based selection within such systems has led to parasitic side reactions, prohibiting long-term study of population dynamics. A prior study employing continuous in vitro evolution gave rise to RNA enzymes that were resistant to a toxicant, although unlike the prey, the toxicant was not itself subject to evolution. The continuous coevolution of RNA enzymes provides a framework for the simultaneous evolution of multiple molecular species. This development, in conjunction with a better understanding of biochemical parameters required for continuous evolution, will enable greater complexity to achieve future molecular ecology studies that employ diverse catalytic motifs. So I think that's a good summary, and I think, understandably, those not in the field might not see the relevance of this type of research. But not only do I think this research is super cool, but I think it is super relevant and a really powerful way to study evolution. They're studying evolution in a test tube, and they're arguably studying it like not in exactly real time, but like pretty close to it, like real time adjacent, and like they're seeing species go extinct, but in this case their species are RNA enzymes. And they're seeing, competition play out. They're seeing coexistence play out. And so, yeah, a really powerful way. Oh, Athens, super cool. And yeah, GG for sure. And so I really appreciate that this was sent by a community member. And like, yeah, yes, anyone wants to join the Discord and ask questions, just research by all means. And this was published in 2009, and I'm curious. I have to assume that they've should have looked before. I have to assume done some sort of follow-up. Oh, I can't click it. Oh, that's like one of the most recent. Oh, maybe not. Okay, well, this person's still around. But yeah, I'm probably gonna have to look up some of the stuff that they've done since this, because yeah, this goes back 1997. We have this Gerald F. Joyce person, and they're still studying continuous in vitro evolution of catalytic function. Okay, in 1997. And then we have this other one. This is 2007. Also continuous evolution. And then, like, yeah, now you have coevolution, still RNA enzymes, and it's 2009. They've been doing this research for a while. So, assuming they're not retired, which maybe they are, but I have to assume that they're probably still carrying out this research. Might have been a follow-up on this 2009. Fine. But with that, that's pretty much everything that I want to cover about this page. I super appreciate that it was, because yeah, it's super cool. Well, I think so. And I think a really nice introduction to get sort of acclimated with me using a tablet and sort of like drawing things out. Although whether it's successful or not in getting the concepts across, I don't know. I can't say. But I'm going to try to incorporate this more and more. Like, hopefully, where I can improve. If you also want to join the Discord and criticize my methods or see where I can do better, that's also an option too. So that's pretty much. I have other stories, but they take too long to cover. So I'm gonna save that for Tuesday when I plan to stream next. They have a bunch of science stories about the food industry, paying influencers, etc., and a whole bunch of other science stories to cover. But with that, I think I'm gonna end this stream here. Let's see if I can find someone to raid. Believe. Yeah, slightly caffeinated is streaming, so I'm gonna raid them. Up. I want to thank everyone that passed by. Time Scroll, thank you so much for passing by. And the follow, super appreciated. And thank you, Elwal Links, for the Prime. That's, yeah, I'm at a loss for words. That's super generous and appreciated. Thank you, Discount Seth Rogen, for the sub. Thank you, Rafa is God. Thank you, Bob Marley. Thank you, Bill, and anybody else that I forgot. I apologize. Thank you, Doffy. Appreciate it. Thank you, Cernu. Yeah, we're gonna raid into Slightly Caffeinated. They're a science streamer, but right now they're gaming. But they're an immunologist, so give them a follow if you want to support more science. I believe they were doing microscopy earlier. I plan to be back on Tuesday. No. Oh, and Wicked Beta, thank you so much for passing by. Super appreciated. I apologize if I forgot you. I absolutely know homozygote. The molecular biologist. They cover a lot of COVID research and some climate research. I totally appreciate. And I've been on a panel with them before, so totally fully acquainted with homozygote. Yeah, they're super cool. And I appreciate their content as well. And arguably the go-to person on Twitch, COVID research. And actually, I'm preparing a stream devoted to long COVID as requested by a community member. So I'm putting that together. And I'm hoping in the next week or two to do a deep dive on long COVID, or at least, some of the research, current research on long COVID. Obviously, that's an emerging area of research that's, evolving, to use the appropriate term. But yeah, I'm planning to put that together and do a deep dive on that and try to present what we know about long COVID and arguably more of the what we don't know. We're still trying to figure out. Yeah, so we're gonna raid into Slightly Caffeinated. Thank you, everyone. Super generous. Super appreciated. And like, yeah, until next time, take care, be well, and be kind. And follow Slightly Caffeinated.
How was your stream? Let me give you a little shout out here. So Corey, let's see. I'm guessing you were doing science and technology. Wait, what? Wait, are there multiple? Wait, I got 2 raid messages.